Got a pretty decent turnout for our first lecture. Glad to see that. Can everybody using the participation thing raise your hand to say if you can hear me? All right, either some people have stepped away or getting most of the hands raised. So again, please raise your hand if you can hear me okay. Again, you're welcome to use that raised hand icon to get my attention during the lecture, or you can, let me get the chat window up. Where's the chat window? Last time. Oh, there it is. Perfect. All right, I got the group chat up on that side, and I got you guys set up here. So hopefully that works. All right, excellent. So I think most people, except for a couple, haven't raised their hand yet, but hopefully that means you guys can hear me okay. So what we're going to go ahead and do is get started with the lecture. So again, here I've got on the front page our game plan for today. We are going to start our discussion of the respiratory system. Sorry, of the respiratory system. We're going to start first talking about the anatomy, and then once we understand the anatomy, then we will get the opportunity to start talking about the physiology and how it works. For this section, there are 3 assignments that I want you to work on. Those are going to be the Unit 22 review and the Unit 23 review. As I was saying during the practice lecture, for some of you who were there, I do want you to do these activities, but you are not, and you will be turning them in, but you will not be turning them in by email, at least not yet. I want a better understanding of how long we're going to be doing this. So rather than getting 100 emails from everybody with all their assignments, I'll either set up a Dropbox and we'll turn them in that way, or I'm hoping we'll be back and we'll turn them in before the exam. So one way or another, they will be due before the exam. So just make sure you know that. There is also a PhysioX. Obviously, if we're not back in the classroom, we're not going to have the opportunity to do wet labs, but the PhysioX is a great lab simulator. So it's a great way to see some of the activities and discuss some of the concepts. So that is going to be important for you guys to do as well. All right, any questions on that? All right, excellent. I will take the stunned silence as a no questions, and let us go to the lecture. All righty, excellent. So again, here is our lecture slides. You guys have these, so you should be able to follow along with that. I've made that stuff available for you. When we talk about the respiratory system, some of the confusion that comes along with that is what we actually mean when we say respiration. Obviously, when people think of respiration, one of the first things they think of is just the process of just breathing in and breathing out, that big type process. That is our pulmonary ventilation. And so, or the mechanical ventilation would be another term for that as well. And so obviously that is clearly one thing that we think of when we think of respiration. However, the whole reason we are bringing our air in and out is to facilitate our external gas exchange. One of the important things to remember about external gas exchange is it is the exchange either between 2 mediums. Those mediums would either be the air. Where's my doodle thing again? There we go. Bring that up. So again, we can think of this in terms of mediums. And in that case, it would be between the air and the blood that we're exchanging these gases between. Or we could think of them in terms of the structures where this external gas exchange is taking place. And that would be the air, of course, would be in the lungs, although we can be more specific and say alveoli. And then, let's spread that out a little bit more. Between the lungs and our pulmonary capillaries. So again, when we think of external gas exchange, it is exchange between us and the outside world. And again, we're exchanging gases between the air and the blood, and between the lungs and the pulmonary capillaries. You can think of it either way. And of course, what are the gases that are being exchanged?
Oh, yeah. What was your weird dream about?
Oxygen and
Oxygen.
dioxide.
Oxygen is definitely one of them, and the other one?
Carbon dioxide.
And carbon dioxide. Excellent. Again, I would encourage to remind everybody that if you have other things going on in the room, you might want to mute your microphone until you have a question that you want to ask or an answer you want to give. Excellent. So those are the gases that are being exchanged between the air and the blood, between the lungs and the alveoli and the capillaries. And another important part about the external gas exchange is which direction are the gases going? For oxygen, for instance, right? Is it coming from the air to the blood or from the blood to the air?
Air to the blood.
Excellent. It's going from the air to the blood. And then, of course, CO2 is the opposite. It is going from the blood to the air. Or again, we can replace those with from the pulmonary capillaries to the alveoli, or from the from the alveoli to the pulmonary capillaries. So we could talk about those that way as well. And that is our external gas exchange. Oops, hold on. Mouse back there. There we go. Excellent. Of course, we already talked about the cardiovascular system, and we talked about the important role it plays in transporting those gases. Once we get that exchange that takes place in the lungs, we need to move them to all the cells of the body. And once those gases get to the cells of the body, that is when our internal gas exchange takes place. And again, with internal gas exchange, again, we are moving blood between, again, either mediums or structures. In this case, the mediums would be our systemic, would again be blood. And in this case, the interstitial fluid. And then our structures that we would be exchanging would be the systemic capillaries and our tissues. Right? And again, it's still those same gases, oxygen and CO2. Only this time the directions are different. Here, our oxygen is going from now the blood into the fluid. Whereas at our CO2, that moves from the interstitial fluid to the blood. Oh, and I still get one letter just like you guys. There we go. So again, we are getting that exchange of gases with the external gas exchange. I can do that with our, there we go. With our external gas exchange, remember it's with the outside world to our cardiovascular system. Internal is from our cardiovascular system to the tissues. That's why it is taking place on the inside of our body. All right. So all of these are things, when we talk about processes of respiration, that is what our respiration is. It all entails all of these things. We often just think of the pulmonary ventilation, but really it's all of these that are taking place. There we go. And then, of course, the whole point of all of this is that cellular respiration. Let me go ahead and erase that. Anybody remember way back to 4:30 what our cellular respiration is? Again, if you're too shy to speak, remember there is a chat window where you can type things there as well.
Aerobic and anaerobic.
True, absolutely. And so let's talk first about the aerobic. What would aerobic respiration be?
including glucose?
Yeah, absolutely. So absolutely, our goal is to use glucose, and we want to break that glucose down. But what do we need to break that glucose down?
Oxygen.
Oxygen, absolutely. The whole point of that breathing in and out is so that we can use oxygen to completely and thoroughly break down our glucose and do that as efficiently as possible. And when we do that, as a result of that, we get a couple waste products. We get water as a waste product from that. And then we also get our CO2 as a waste product. Oops, wrong button. CO2 as a waste product of that, which is why we then have to exhale that CO2, right? This whole process is why we breathe in oxygen and breathe out CO2. And the whole reason we do this is so that we can get the energy necessary, and that energy, of course, is primarily used to make ATP. So that, and of course, where does this complete breakdown of glucose with oxygen take place? What organelle of the cell? Where in the cell? What organelle?
Mitochondria.
Yeah, in the mitochondria, absolutely. So it's the mitochondria where this takes place. All right, so this is everything that is involved in respiration. I wonder what happens if I change the slide now. Oops. Oh, and there's the pretty picture that goes along with it. So let's go ahead and erase that. So again, this shows many of those processes here where we see the external gas exchange, the transportation that is taking place, the internal gas exchange, all of those things that we're talking about. And then, of course, we fill the alveoli with our pulmonary ventilation, and our cells then use that oxygen to make energy and give off the CO2 as a waste product. All right, excellent. So again, when we talk about respiration, we have our pulmonary respiration, our ventilation, the mechanical movement where we use our muscles to be able to change the volume, and that change in volume changes the pressure, and that allows us to, excuse me, and that allows us to move air into and out of our lungs. Oops, too much. As we talked about, the external respiration, or gas diffusion, that is where we move the gas from inside out, again, between the alveoli and the pulmonary capillaries. And we already talked that in this external gas exchange, or the external respiration, oxygen goes from the alveoli to the pulmonary capillaries, CO2 from the pulmonary capillaries to the alveoli. And again, we could interchange those with air and blood, if that helps you to remember that it's external. And then our internal respiration, again, is between our systemic capillaries. Again, remember when we talked about our cardiovascular system, one of the things we talked about is the difference between our pulmonary circulation and our systemic circulation. Our systemic circulation, which is most of the blood vessels, the arteries are oxygen rich, the veins are oxygen poor, and we talked about all of that. Whereas we know our pulmonary screwed things up that way. The arteries were the oxygen poor, going to get the oxygen, and then coming back in the veins, oxygen rich. So we have those. And again, same gases, oxygen and CO2, but they move in a different direction. With the internal, oxygen leaves the systemic capillaries to the tissue. CO2 leaves the tissue into the systemic capillaries. And with the chat window up, I can't see what that says. Cellular respiration. We did that as well in the mitochondria. So excellent. We did that on the previous page. So when we think about the respiratory system with all of these types of things that are going on, really what we're primarily going to be focusing on is parts 1 and parts 2, pulmonary respiration and external respiration. Right? We need to understand the numbers involved in this process, but what we are going to need to do is focus on the first 2 parts. Obviously, cellular respiration we talked about in 430. You'll get it in micro if you haven't taken it already. And then we already kind of talked a little bit about capillary exchange in the cardiovascular system. All right. Our respiratory system's functions, as we talked about, are to move and filter air, get that air mechanically into and out of our lungs, and then have that external gas exchange that is going to take place. But we got to remember that our respiratory system does other things for us. In our nasal cavity, we have that first cranial nerve, that olfactory nerve, dangling down into our olfactory epithelium through those cribriform plates in their olfactory foramina of the ethmoid. All things that I'm sure sound familiar to you in a vague sense, hopefully, because we talked about that in 430. And so that sense of smell, by the movement of those chemicals dissolved in the moisture of our nasal cavity, allows us to sense our sense of smell. But one of the other important functions of the respiratory system is our pH regulation. Our respiratory system and our movement of air is controlled dramatically by the pH of our blood. As we talked about in 430, we talked about proteins and their structures and how they're enzymes, and those enzymes are necessary for pretty much every chemical reaction that takes place in the body. And as proteins, they're sensitive to temperature changes and pH changes. Right? There's something magical about our cardiovascular system. It is involved in the movement of gases. But one of the important things to remember, or to learn if you didn't already know this, is there's something that really magical that happens when carbon dioxide mixes with water. When carbon dioxide mixes with water, does anybody know what we get as a result of that?
Bicarbonate.
Well, true, bicarbonate, but first, actually what we actually get is an acid. That acid happens to be carbonic acid, but you are absolutely correct. That carbonic acid is then going to rapidly dissociate into our hydrogen ions, and then also bicarbonate, which is HCO3 negative. Bicarbonate. Let's just write out bicarbonate because that looks like liquid. And that bicarbonate is an important pH buffer. So basically what happens is the more CO2 you get in your blood mixing with your water, the more acidic your blood gets as a result of that. I know a lot of times when we think about respiration, we think about holding our breath, right, when we don't get our way, right? I don't want to go to school today, and so what do? You hold your breath. And your wife says, tough, you have to teach today, get upstairs to your room. But when we try to hold our breath, eventually we have to breathe. The reason you breathe isn't that you run out of oxygen. The reason that you're forced to breathe is actually you build up too much CO2. Your blood becomes too acidic, and that forces you to breathe so that we can exhale that CO2. As we'll talk about and as we'll see, CO2 is a much more important factor in determining the rate and the depth of which you're taking a breath than actually oxygen levels are. And we'll get an appreciation for that when we actually see the numbers. You know I'm not huge on numbers, but there are some numbers that are important. And the partial pressures, because when we talk about gases dissolved in water, that's what we talk about is the partial pressures. The partial pressures of oxygen and CO2 on its journey through our body is going to be numbers that we are going to be responsible for. All right, so let's clear that. And oops, no, wrong button again. All right. Lastly, obviously, the whole way we are able to have this lecture in the format that we do is then the other function of the respiratory system is to force air across my vocal folds to cause them to vibrate. I then am able to use my mouth, my oral cavity, my nasal cavity, my lips, my tongue, my teeth to shape that sound to produce speech. So that ability to produce sound from our respiratory system, whether it's speech like this or whistling or any other kinds of functions, is an important function of the respiratory system as well. All right, I'm gonna grab my water. All right. When we talk about the respiratory system, not shockingly, not surprisingly, the way we classify pretty much everything is anatomically and physiologically. So when we talk about any of the systems, we can talk about them from their structural standpoint or from their functional standpoint. And the respiratory system is no different. In our respiratory system, it's basically divided both anatomically and from a physiological or functional standpoint. From an anatomical standpoint, basically, as you can see from the illustration, we have two components to our respiratory system. We have the upper respiratory system, which, as you can see, is basically from your Adam's apple or your laryngeal prominence, as we'll talk about, up to the top. And that is our upper respiratory system. And then from your Adam's apple basically down is what we call the lower respiratory system. Right? Everybody's been talking about the lower respiratory system because of the coronavirus right now. So when the coronavirus gets into your lower respiratory system, you get short of breath, you have the coughing, all the problems and everything that goes along with that. This division anatomically, I guess, is useful from a, I guess, maybe clinical standpoint, because you have to basically either deal with things that are the head or deal with things below the head. However, it really isn't that meaningful. For me, I find it to be much more meaningful to divide the respiratory system functionally. And when we divide the respiratory system functionally, there are basically two main components to it. The first is the conducting zone, and the second is the respiratory zone. The conducting zone, as the name indicates, is the portion that is involved in the movement of air, getting air into and out of our lungs. However, it's more than just the movement of it. It is also the processing of that air as well. As we talk about, as we'll mention when we get to the lungs, our lungs have a massive surface area. One of the reasons for that, as we will talk about, is our gas exchange, as vitally as important as it is to get oxygen into your body and CO2 out, right? How long can you survive when someone puts a bag over your head, right? Not very long. It is vitally important to get air into and out of our bodies. But that entire process, those entire gas exchange, that external gas exchange, is a completely passive process. No ATP is used in it at all. So one of the things we need to do is try to make that process as efficient as possible. One of the ways we do that is by having a huge surface area. When we have that huge surface area, that is going to help to facilitate diffusion. And so, as they always talk about in the textbook, the surface area of your lungs is close to the size of a tennis court. But the other thing we need to do then is to process that air. And so by processing it, I mean, there's three main things we need to do. We need to clean it, because again, Sacramento air has a tremendous amount of pollution. You don't want to just take a big whole handful of it and stick it right down there into your lungs. But the other two things that we want to do to it is, first, we want to moisten it. Gases diffuse much more rapidly in solution. So we're going to moisten that air. And the other thing we're going to do when we're dealing with passive processes is we want to increase the temperature. So we want to warm it, because the warmer things are, the faster they move. So we want to moisten the air, we want to warm the air, and we want to clean the air. And that is the function of the conducting zone. This helps us to understand also why the oral cavity is not a part of the respiratory system, right? We didn't get a chance to talk about the digestive system, but when we do get a chance to talk about the digestive system, one of the things that we'll talk about is that, while theoretically this morning with breakfast, you could have put a straw up your nose and you could drink your glass of milk that way, just because you can use your nose to get milk into your stomach doesn't make your nose a part of the digestive system. And it's the same thing for the respiratory system. With the respiratory system, anybody who's ever had a cold survived that cold because they were able to get air through their mouth into their lungs. But just because air can move from your mouth into your lungs doesn't mean that it's part of the respiratory system. Yes, it is a pathway where air can move, but unlike the nasal cavity, the oral cavity is not specialized to clean, to moisten, to warm the air the same way the nasal cavity is. And since it's just a conduit in that process and doesn't process it the same way, it's not considered a part of the respiratory system. So our goal of the conducting zone is to get the air into the lungs and process it, and more specifically into the alveoli, because in the alveoli is where our respiratory zone is, and that respiratory zone's function is the gas exchange. That is where we're getting the movement of gases, oxygen and CO2, into and out of our body. So while both the anatomical and the functional are ways of describing or dividing the respiratory system. Like I said, I think the functional one of the conducting zone and respiratory one is a more meaningful division. All right, questions on that? All right, here's the pretty picture that goes along with this. Again, when we're talking, oh, knocked over my iPad. That's never a good thing. All right. Here again, from an anatomical standpoint, we see the structures of the superior or upper respiratory system. Again, we have the nasal cavity. It starts with the external nares. It then enters into the nasal cavity, and we'll talk about some of the nasal cavities. Anybody remember what these bumps are called? These are the nasal meatuses. This is the concha, sorry. This is the inferior nasal concha. Remember, it is its own bone. This is the middle. Can you guys see the little light up thing when I'm doing that?
Yes.
Okay, excellent.
Yes.
All right, so so this again is the inferior nasal concha. It is its own bone, one of your facial bones. And then remember these two, the middle nasal concha and the superior nasal concha, are bone features of the ethmoid bone. So the conchas are the bumps, and then we have the meatuses, which are the small airway passages underneath them. And I have a great picture that shows this we'll take a look at in just a minute. We see the framework of the nasal cavity with the sphenoid bone and the nasal bone and the frontal bone and the hard palate, which is the maxilla and the palatine bone, all of those things. They then lead to the internal nares. So the external nares is the external opening. This is the only place the opening from the outside world feeds into the vestibule. That's where your finger goes when you pick your nose. Then when we leave the nasal cavity, we leave through the internal nares. The internal nares is basically formed by the sphenoid and the palatine bones that form an opening there that allow us into the pharynx. The pharynx, of course, has three main regions: the nasopharynx, the oropharynx, and the laryngopharynx, which are basically identified by the openings that they are associated with. We saw some of this on the last test, where we saw the tonsils, like the pharyngeal tonsils that are at the top of the nasopharynx, the palatine tonsils, which are in that fosse. Here we actually see one of those arches. It looks like this one is the palato-esophageal arch, because it's going back towards the pharynx. And then the lingual tonsils on the back of the tongue. And then from there we lead into the pharynx, and then from the pharynx into the larynx. And as we'll talk about, the larynx is where, among other things, we have the Adam's apple. And again, we talked about this when we talked about our tissues in 430. This is the epiglottis, one of the two structures that are made up of elastic cartilage that allows us to bend over and see that movement of food into either the esophagus and allowing air into the airway. All right, questions on that? All right, excellent. There we saw it in the illustration. Here we see it in a sagittal section of the cadaver, where again we see those same structures. Notice the other thing that you can see nicely about this, let me bring my spotlight back, is we can see one of our four bones that have a paranasal sinuses. There are the paranasal sinuses, which are the air-filled locations and regions associated with it. The sphenoid is one. The frontal bone has a sinus. The ethmoid bone has all of these sinuses inside of it, and the maxillary bone. Those four bones all have air-filled sinuses and form what we call our paranasal sinuses. Those sinuses and these concha basically do the job of producing turbulence. We don't want to take the air and bring it straight down into our lungs. Like I said, we want to process it. We want to clean it. We want to warm it. We want to moisten it. And so the way we do that is by swirling the air around. The air is going to swirl around inside of these spaces. This also is what brings it up to the olfactory epithelium so that we can smell. but it allows it to be processed on its way down to the lungs. And so those paranasal sinuses help in that process. All right. So again, I've done some of this, but we'll go through it all together. The nose is the only external respiratory structure of our entire respiratory system. As I mentioned, air enters through the external nares, which are your nose holes, and they enter into the connective tissue. Notice there is a rubbery fibrous connective tissue vestibule, which is, like I said, the flexible part of your nose, right? We know that we have the very top of your nose, which is your nasal bone, and then we have that hard, rigid hyaline cartilage, which basically forms the bridge of your nose. But the rest of your nose is this kind of rubbery, flexible connective tissue. And that's basically where the vestibule is. As that previous picture showed, let me see if I can go back. As we can see here, notice there are these long hair-like structures that are known as the vibrissae. And those vibrissae are your nose hairs, right? Those are the ones that basically stop large dust and debris, small insects, and things like that from flying into your nasal cavity. From there, as I said, the nasal cavity is comprised of bones like the maxilla, the palatine, the sphenoid, the frontal bone, all of those bones. And as we learned in 430, what is the epithelial tissue that lines our nasal cavity? Well, it's very easy. What membrane lines our nasal cavity?
Mucus.
Yeah, it's a mucous membrane, absolutely, because again, mucous membranes line what are open to the outside world. And so while that is our mucous membrane in there, what is the epithelial tissue?
Stratified cuboidal.
No, remember stratified, it looks stratified, but it isn't actually stratified, right? It's our...
Stratified squamous.
No, that again is going to be in our oral cavity. Now in our nasal cavity, remember our nasal cavity is one of two places where we find our ciliated pseudostratified columnar epithelial tissue. Remember that ciliated pseudostratified columnar epithelial tissue, its job is to, as part of a mucous membrane, our mucous membrane makes mucus, and then the cilia in our nasal cavity move that mucus back towards the back of the throat, or really back towards the nasopharynx, at which point that mucus can either then be swallowed or spit out. Right? Obviously, the advantage of spitting out the mucus is that any dust and debris, any harmful pathogens that are in it can then be expelled from the body that way. So why would we swallow it?
Because acid will take care of that too.
There you go, absolutely, because as we talked about, one of our body defenses is our stomach acid. That stomach acid will denature most of those harmful pathogens and again, stop them from getting inside of our body. All right, unless, again, you've been very bored with your time at home and so have been snorting massive amounts of cocaine, you should not be able to take a handkerchief, stick it up one nostril, and pull it out the other side. And the reason for that is we have a nasal septum that divides our nasal cavity into two halves. There is some cartilage and some fibrous connective tissue that forms that nasal septum. But we also have two bones and a bone feature that form it as well. Anybody remember what the bones and bone features that form our nasal septum are?
Palatine process, or?
No, that's more the roof of the mouth. So the nasal septum, remember, that divides it in half was the vomer. That kind of, exactly, the perpendicular plate of the ethmoid and the vomer bone. So those two things there are what form our nasal septum. There was an interesting study. One of the interesting things about this is, again, remember, as we talked about, gas exchange is passive. We always have to remember that. So we want to maximize the efficiency. And just depending on what's going on in your particular environment, if it's particularly dry or things along those lines, one side of your nasal cavity may not be as ideal. Maybe it's more dry. Maybe it's a little cooler in temperature, so it's not processing the air as much. So inside our nasal cavity, we actually have erectile tissue similar to the erectile tissue in the penis of a male and the clitoris and the labia of the female. And what can happen is if one side becomes less ideal, that erectile tissue can swell and can constrict the opening, right? At any one particular time, if you cover one nostril and try to breathe in, you might be able to breathe in better on one side than the other, because it'll alternate back and forth to try to maintain the efficiency of that processing of the air. And I think it was like 6 or 8 years ago now, there was an interesting study where a group of researchers looked at cognitive abilities in individuals. And they claimed, although I've never seen anybody recreate their data, that, for instance, people had different math skills, different competencies in math when air was going in their right nostril versus when it was going through their left nostril. So I'm not sure how exactly the air coming through. Maybe there's more gaseous fusion through the cribriform plate, so maybe you get more oxygen to the frontal lobe on those, one side versus the other from that. I'm not sure exactly what they claim the mechanism by which there was a difference, but they said that statistically they did show a difference in math competency when air was going in one nostril versus the other. So again, it's just an interesting fact from that. And again, as we talked about, again, anybody who's ever had a cold survived it because they could get air through their mouth, but the mouth is not considered a part of the respiratory passageway. The same way you could drink your glass of milk in the morning by snorting it through your nose and getting it into your stomach, but that doesn't make your nose part of the digestive system. Here, oh yeah, I do have the pretty picture. As we talked about, there are those paranasal sinuses. Here we see them again. There's this great picture from your textbook where we see the 4 sinuses. So again, we have the frontal sinuses in the frontal bones, those ethmoid air cells inside the middle and superior nasal concha, that sphenoid sinus way to the back, and the maxillary sinuses. And again, these sinuses, along with the conchas themselves, which I believe, there's the cool picture. I love this picture. You get a posterior view of a coronal section. So the rest of the face is out this way. And so as you can see, yep, it is gross. As you can see, here's that nasal septum, that vomer bone and the perpendicular plate of the ethmoid. And then here we can see the superior and middle nasal conchas. We can see the inferior nasal concha, which is its own bone. And then notice we see the spaces underneath them. Those spaces underneath them, as I mentioned, are the meatuses. So the point of all of this, and again, there's the frontal sinus, there's the maxillary sinus. We see a little bit one of the air cells of the ethmoid. So the only one we don't see is the sphenoid because it would be more posterior than this in its view. The point, as we talked about, is to produce that turbulence, to swirl that air around, and to when we swirl that air around, give it the opportunity to be processed, to warm up, to get clean, and to moisten. And again, you might not have thought of it in these terms, but anybody who's been outside on a cold day is aware of it. Because when you breathe in, the air coming into your mouth is completely clear and you can't see it. But when you breathe out, you can see it. And the reason you see it is because you've warmed it and you've moistened that air. And so as that warm, moist air leaves your lungs, exits your mouth onto the outside world, the cold condenses the moisture. And so you see your breath when it's cold outside because of that warming and moistening of the air that takes place. All righty. So again, there's the magic word. It produces that turbulence to, again, process that air. And that's why the oral cavity is not considered part of the respiratory passageway. It doesn't produce that same turbulence. It's not specialized to process air. From there, as we mentioned, it is going to leave the nasal cavity through that internal nares. This is usually when I would take a skull and show it to you on the OMO so we can actually see this. But there's some good pictures on your textbook where you can see that. And again, if you think back to 430 when we were learning the concha, if you remember, when we looked from the inferior side of the skull past the hard palate, back here, this is where we were able to see all 3 of the concha. So basically that opening you were looking through to see all 3 of the concha, that is that internal nares. All righty, questions on that? All right, excellent. I have been talking nonstop for 40 minutes. I know normally we go about an hour, but I think things are going to go a little faster for this. So let's take a quick 5-minute break. I'm going to pause the recording so that when we go back and look at this, we don't have to see it sit here for 5 minutes and do nothing. So I'm going to pause the recording, take a quick 5-minute break, stretch your legs, get something to drink, go to the bathroom, whatever you need to do. And we will meet back here at 8:45. So at 8:45, I will be back on here. So thank you for catching that. All right, so let's start again. So like I'm saying, we have this ciliated pseudostratified columnar epithelial tissue in our nasal cavity and the nasopharynx. Our oropharynx and laryngopharynx, which are passageways for both food and for air. They have that non-keratinized stratified squamous epithelial tissue. But once we're back into the larynx and the trachea, we're back to an air-only passageway. And that air-only passageway, again, will be aligned with our ciliated pseudostratified columnar epithelial tissue. And those cilia are sensitive to things like cold. They can be overworked over in that way. But the other thing that can damage them is smoking. Smoking damages the cilia. And as a result of that, and again, while you may not have thought of those terms, if you know someone who's a chronic smoker, then one of the things that you know is that their cilia don't work properly, so they can't move the mucus and dust and debris out of their trachea. So instead, and I apologize for this in advance, you get that kind of smoker's cough. And that cough is basically how they have to dislodge and try to expulse that mucus that is being produced. One of the concerns for individuals who are chronic smokers is that when they then get a cold on top of everything else, what can happen is they take cold medicines, which typically have cough suppressants. So now their cilia aren't working because they're smokers. Now they're taking a cough suppressant so that their cough reflex doesn't work. So instead, all that mucus, all that dust and debris that's in their trachea drains down into the lungs. So typically individuals with smoking, especially when they get the cold, tend to have more complications, more chances of COPD, more chances of other types of respiratory distress, more chances of pneumonia or other type of respiratory infections. And then also, as we've talked about, smoking, well, not talked about, but as we know, smoking is also associated with lung cancers as well. And a lot of that is that inability to move that dust and debris out of the lungs. Here we have the pretty illustration of this, of these ciliated pseudostratified. Again, remember, it looks like it's multiple cell layers, but it isn't actually. It has those motile cilia on top, and again, their goal is to move that mucus up, away from the lungs, up towards the, really the laryngopharynx, where again, it can either be swallowed down or spit out. Of course, it is a mucous membrane. So we have our epithelial tissue. Oh, actually, let's go back. So we have that, here we'll go all the way back. So ciliated pseudostratified columnar epithelial tissue in the nasal cavity for following the pathway of the air. Then into our nasopharynx, our oropharynx, which again is a non-keratinized stratified squamous, through the laryngopharynx into the larynx, ciliated pseudostratified columnar into the trachea, from there into the lungs. Now, as it enters the lungs, it at first is still that ciliated pseudostratified columnar. However, it is going to transition. Because if you think about it, when we finally get down to the alveoli and we want our gas exchange to take place, since it's passive, does not use ATP, not only do we want a massive surface area, but we also want it to go the shortest distance possible. We already learned in the first part of the class that our capillaries are a single layer of endothelium, a simple squamous epithelial tissue, one layer of flat cells, where our alveoli needs to be one layer of flat cells as well. So what happens is we transition from a ciliated pseudostratified columnar to a simple columnar to a simple cuboidal. And then finally, when we reach the alveoli, it is going to be a simple squamous epithelial tissue. So we have, what is that, close to a half dozen different types of epithelial tissues lining our respiratory passageways. However, being a mucous membrane, all of those different types of epithelial tissues sit on top of just one type of connective tissue. And of course, what is that one type of connective tissue?
Areolar.
Areolar connective tissue. And what do we call an areolar connective tissue that is part of a mucous membrane based on its location? What's that magic term?
Lamina propria.
There you go, lamina propria. Excellent. All right, so we have that areolar connective tissue. And again, remember, if you don't want to have to speak out loud or don't have a microphone, you can always type in the chat window as well. So that's always a way you can answer me as well. Although I do appreciate the people who interact with me, so then I don't feel like I'm just talking to myself. I don't know how people do radio shows this way. I like the interaction of the classroom way, way better. All right, so we have that areolar connective tissue that, based on its location, is that lamina propria. Excellent. All righty. No, I want that back. There we go. Have that. And then we do that. There you go. Now, again, in our upper respiratory tract, especially in our nasal cavity, especially in our trachea, we have a massive number of major mucus glands. Again, that mucus is going to do two things. It's sticky, so it is going to capture that dust and debris. And of course, remember, our glands, like the goblet cells that make mucus, don't just produce mucus. They actually produce a protein called mucin, M-U-C-I-N, and that mucin then, when it moistens with water, becomes mucus. And so again, it captures the dust and debris and helps to moisten the air. Remember, the other job of our respiratory passageway is to warm the air. And of course, as we talked about in the cardiovascular system, our blood is 2 to 3 degrees warmer than our core body temperature. So by having a large number of very superficial blood vessels in our nasal cavity, we can bring that warm blood close to the surface to warm the air up, make the molecules move faster, and that's going to facilitate better diffusion. Of course, the problem with having a whole bunch of superficial blood vessels really close to the surface inside your nasal cavity is that you take an unexpected blow to the nose, right, or get a little too vigorous picking a booger, and as a result of that, you can damage one of those blood vessels and you get that nosebleed. And again, it could be dry air, it could be, mechanical abrasion, it could be any number of things that can cause that injury. And if you get chronic blood nosebleeds, often that is one troublesome blood vessel that often causes that problem. And so what they can actually do is go in and cauterize that. And since you have plenty more, you'll still be able to warm your blood, but they can stop that one troublesome blood vessel from constantly breaking and causing those chronic nosebleeds. Again, remember also at the top of the nasal cavity, we have our olfactory epithelium. We have the cells where those neurons, our olfactory neurons, are located. Remember, they're bipolar neurons. We learned about that in 430. Remember, we also learned something interesting about those bipolar neurons. They're some of the few neurons in the body that regenerate fairly rapidly. As we talked about in 430, at the time when that was found, that we actually had neurons that regenerated, that was a huge, huge big deal, because normally we think of neurons as not being able to regenerate. So a tremendous amount of time and effort and money and research went into studying these olfactory neurons to try to figure out what's so special about them. Can we do something special to the other ones that will make them regenerate as well? Because then that could help with, recovery from spinal cord injuries and things along those lines. And unfortunately, the research never really went anywhere. Research with stem cells was much more successful, and so that's where most of the money in the research has gone. But we do have those bipolar neurons that do allow us to regenerate and have that sense of smell. So, as we mentioned, when we leave that internal nares, the air enters into our pharynx. Our pharynx is what we commonly think of as our throat, which is just a big muscular air passageway. Come on, There we go. As we talked about, there are three regions of it, basically divided by the openings they are adjacent to. So we have our nasopharynx, which is open to the nasal cavity. And again, the nasopharynx is lined with a ciliated pseudostratified columnar epithelial tissue. The nasopharynx is also where we found our pharyngeal tonsil. The oropharynx is the portion of the pharynx, of the throat, that is open to the oral cavity, lined with a non-keratinized stratified squamous, as is the laryngopharynx, also lined with a non-keratinized stratified squamous epithelial tissue. Because again, food and drink have to pass through these as well. And here we see it with the words. Come on. Here we see it with a pretty picture. So again, what we commonly think of as the throat is this pharynx. We have that nasopharynx for air only, oro and laryngopharynx for both air in its passageway and food in its. One of the few disadvantages of doing the respiratory before the digestive is in the digestive, we'll talk about the swallow reflex of how it is that we move these structures to get the food into the esophagus. But as you can see from the illustration, the default setting of this structure is up so that the air can flow in. No, great question. So just the oropharynx and the laryngopharynx are lined with the non-keratinized stratified squamous epithelial tissue. The nasopharynx, like the nasal cavity, is lined with the ciliated pseudostratified. And the way I remember it is when we're talking about the upper respiratory tract, which again is basically from the Adam's apple up, the things that are air only, the nasal cavity and the nasopharynx and the larynx, those things are ciliated pseudostratified columnar, because all they have to worry about is the air, so they want the mucus, they want the cilia to move the mucus. The oropharynx and laryngopharynx are part of the pathway for food. So not just air passes through this, but hot chocolate, hot coffee, acidic wine, sharp tortilla chips. So this portion, it's more important that we protect it. So just the oropharynx and laryngopharynx are lined with the non-keratinized stratified squamous. Great question. Thank you for asking that. And there's a pretty picture for it again. All right, as I mentioned, the job of the larynx is to route the air and food into the proper channels. I know we haven't had the digestive system, so it's a little bit of a disadvantage, but what you can do is put your hand on your throat, and when you put your hand on your throat, you can swallow. And when you do that, what you feel, first, the bump that you feel is this enlarged piece of cartilage. It's what we commonly call the Adam's apple, but that will be the last time we call it that. We will, of course, use appropriate anatomical terms always for that. But what we do is we feel that Adam's apple elevate and then depress. When it elevates, that elastic cartilage epiglottis folds over the opening of the larynx so that the food and drink gets moved instead into the esophagus. This is why, for instance, if you start to laugh when you're swallowing something, or you start to talk while you're swallowing something, one of two things happen. If you laugh, what often happens is you are forced the air out with that big forceful laugh, and that can cause the food to spit out the mouth, or can actually go up into your nose. Or what happens if you accidentally breathe while you're swallowing, or try to breathe while you're swallowing, that food or drink can actually get into your larynx. In the larynx and also in the trachea, as we'll see, there are a couple areas. The first one is right here, this fold of the mucous membrane that's up here, and we'll get a better view of it in just a minute, is called the vestibular fold. That vestibular fold, it has a highly sensitive receptors. So if some type of irritant, like a drop of water or a speck of dust or a fleck of pepper or something like that gets in there, it causes violent coughing to help to expulse that air and then that whatever that debris is from there. And then way down at the end of the trachea, we have something similar as well. So it is this larynx with its epiglottis that is responsible for directing food and air to the correct locations. As we talked about, its lumen is lined, again, because this is supposed to be just a pathway for air, with a ciliated pseudostratified columnar epithelial tissue, and it is lined with a mucous membrane again. We have several great models of this in the classroom if we ever get a chance to get into it again. However, oh, and this is a good point to bring up, so this is an important note, so pay attention. If you look on our Canvas site, there's a new module that I set up at the top of it, that there is, where I will put links to materials. Cosumnes River College, one of the Los Rios schools, has a great, what they call their visual anatomy lab, where they basically taken pictures of all of the charts and all of the models, and they have presented them for you there. And they use, because not surprisingly, they're a Los Rios school like us, many of the same models, many of the same charts that we do. So you do have access to some of those things. While we won't be in the classroom to learn the anatomy, you do have an anatomy list that you are going to be responsible for. And so being able to recognize it on things like that is definitely going to be something that is going to be important. So if you have not done so already, if you haven't noticed that I put it in there, please pay attention to that and take a look at that. It is a great resource. If you have any more, please share them with me and I will share them with the class. And as I find more, I will post them as well. I haven't had a chance to look around too much. You will be happy to know that while my 430 students were taking their exams yesterday, I finally got around to grading your exams. So we're processing it. I am 3/4 of the way through our lab exams. So I'll have the lab exams done. I've already graded your presentations, and I will tell you right now, everybody did an amazing job. Everybody got at least a B or better on it. So everybody did excellent on those. But then, so I'll finish the lab exams. I'm hoping to have the lecture exam scores done within the next couple days. And I'll get, as soon as I get all those graded, I'll get all your grades updated and posted onto Canvas so that you'll have a chance to see that. I don't have any idea how I'm going to get the exams back to you, but we'll figure that out as we get to it. All right. So, larynx. As I mentioned, there are the two folds in here. One of these folds is a protective fold, and the other one, as we mentioned, both opens and closes the airway, but also is the one that we vibrate to produce our speech. Now, these are often what are sometimes referred to as our true vocal cords and our false vocal cords. Those are not the appropriate terms. We have the vocal fold, because it is a fold in the membrane, and the vestibular fold. And we will talk about and see those in just a second. And has that made a liar out of me? Yeah, see, no, there it is. Good, excellent. Vocal fold, it does say fold. All right, perfect. All right. Now, the larynx, and this is one of the important things that we're going to see, right? Again, we haven't had a chance to talk about our digestive system, but even when you're quarantined at home alone, you are not constantly eating. And so when food is not passing through many parts of your digestive system is a big muscular tube, and that muscular tube basically collapses. It is closed. It's not open if it's not, doesn't have a chunk of food in there. However, our respiratory passageway, aside from the fact that we need to constantly be moving air into and out of our lungs, we also want to maintain that airway open, right? My wife's birthday, despite all the quarantine stuff, my wife has her birthday, and for her birthday, one of the things we did was blow up balloons. When you get a balloon, what's the first thing you do to a balloon when you go to blow it up?
Stretched out.
Stretch it out, exactly, because you're loosening the rubber and it makes it easier to fill it up when you loosen the rubber.
Mm-hmm.
Nope, that is not why you do it. The reason that you stretch it out is what it actually does is it separates the two sides of the balloon from each other. If you just pull a balloon out of the packet and try to blow it up, it is incredibly hard to do. And the reason it is incredibly hard to do is because the two sides are attached to each other. And when the two sides are attached to each other, it's very hard to force the air in and force them apart. However, when you stretch the balloon, that separates the two sides. And when the two sides are separated, it's much easier to fill up. Another great example, and this is an important one we'll talk about moving forward. Again, if it ever starts to warm up again, I thought we were into spring and now we're getting all this rain and everything. But when we get around to summer, which again, thanks to global warming, we will definitely have, you're sitting out on your back porch with your big glass of iced tea or lemonade in that glass, and you're getting all that wonderful condensation on the outside. and it's sitting on your glass outside patio furniture tabletop, right? When you go to grab that glass after it's been sitting there for an hour, is it easy to lift up? No, and the reason is that water has got underneath there, and the two surfaces, basically the magic of water, the surface tension of water, holds that glass to there, so it's hard to lift up. Our airways are very moist, and so that same type of thing could happen. So what we want to do is we want to keep our airways open. And one of the main ways we do that for the big parts is with structure, with cartilage. And that's what we see here when we look at this picture of the larynx. The larynx is your airway that needs to have some flexibility with this epiglottis that's here. But it actually has 8 rigid pieces of hyaline cartilage that is going to hold this airway open so that it doesn't collapse. Because if it collapses, it's going to be really hard to separate it and open it again. And since we're taking breaths basically every 5 seconds, right? So that's 12 a minute or whatever it is. That is a lot of force that we would need unnecessarily to open that up. So we have this rigid structure to keep it open. Now, as we can see here, this big shield-like part in the front is what is known as the thyroid cartilage. It is the largest piece of the larynx, and it also has that prominent characteristic that sticks out that we commonly refer to as the Adam's apple. It is called the thyroid cartilage because, as we saw in the endocrine system, our thyroid gland is that U-shaped gland that sits right here with that narrow isthmus holding the 2 lobes together right in the middle, for those of you who didn't get that right on the lab exam. And this enlargement is what is known as the laryngeal prominence. Everybody has a laryngeal prominence. However, it tends to be more pronounced in males. And the reason it is more pronounced in males is at puberty, when males are producing a large amount of androgens, those androgens, among the changes that take place physiologically in our body, one of the things that does is our cartilage gets bigger and grows more actively as well. And so that increase in androgens leads to an enlargement of that laryngeal prominence. And again, that's why it's called the Adam's apple, because it's more prominent in males. Notice as we look at it from the front, it's this big, huge shield-like structure. And there's that laryngeal prominence sticking out that way. But I think, hold on, that's the picture I want. So there again is that thyroid cartilage. But notice as we look at it from the back and from the side, it only goes 3/4 of the way around. It is really just a C-shaped chunk of cartilage. It's open in the back. So here as we look at it from the back, we can see the other structures inside because it is open. In fact, there is only one piece of cartilage of the larynx that is completely circular, and that is the base of it. The base of it is this structure here. So, oops, wrong button. There's the thyroid cartilage. The base of it is this structure called the cricoid cartilage. Notice the cricoid cartilage is smaller in the front on the anterior side, larger on the posterior side. And here we see that change in size from more narrow anteriorly to more larger posteriorly. But this is the only circular piece of cartilage of the larynx. So it is a rigid structure, but we also still need some flexibility to it. However, where it attaches to our trachea, that is the most restrictive part, because that is the part that is completely cartilage all the way around. All right. As we talked about, there is a 9th, and that's if we go back on the pretty picture. The 9th cartilage is that epiglottis. The epiglottis, as we said, is the only piece that is not hyaline cartilage. Everything else here is hyaline cartilage except the epiglottis. The epiglottis is elastic cartilage, and it gives its flexibility so that it is able to bend down over the top, over the opening. So we have those 3 big pieces. And then we have this interesting structure here. Notice this triangular structure here and here. So we'll put that there. We can see it from the side, is what is known as the arytenoid cartilage. These arytenoid cartilages are actually what rotate. There are ligaments and muscles that attach to it to allow them to rotate. And it is the rotation of that which is what opens and closes your airway. The opening in your larynx where air can escape or come in is what is known as the glottis. Right? That's why the epiglottis is what it is. Epi, of course, means on top of. It is the epiglottis that sits on top of the glottis. And again, I've got a pretty picture that will show this to you guys in a second. But it is the rotating of the arytenoid cartilages which opens and closes the glottis, which is what allows the air to come up through. And as it comes through, it's able to vibrate our vocal folds. Notice on top of the arytenoid, and here I think you see it on the side view a little bit better, this lateral view here, sitting on top of the arytenoid cartilages are these two tiny little pieces of cartilage. Oh, no, that's not the one I want. Where is it? Did it show up? Oh yeah, there it is. The corniculate cartilage. These little nuggets, cornicobs, little corn kernels right on top of the corniculate cartilages, and they help to stabilize the vocal folds, and again, as this arytenoid cartilage rotates. Notice the arytenoid cartilages are paired, the corniculate cartilages are paired, but the cricoid and the thyroid are not. So 1, 2, 3, 4, 5, 6. 6 hyaline cartilage pieces. However, if you remember, I said there are 8 hyaline cartilage pieces that form the larynx. And that's because the other 2 you don't see here. Notice there is one other structure you see here as well. Any idea what this might be?
The hyoid bone.
Yeah, there you go. It says right there if you weren't sure. Absolutely, the hyoid bone. What was so special about the hyoid bone again when we learned the skeletal system?
It doesn't articulate with any other bones.
Yeah, it is the only bone in the body that does not articulate with another bone. It is that movable base of the tongue, but it is held in place just by ligaments, just by tendons, just by cartilage. Notice also there are a lot of additional structures here, a lot of little tiny ligaments and other things along those lines that we can see. When you look at your anatomy list, as I'm sure you have, you will notice that you are responsible for the thyrohyoid ligament. That is one of the ligaments you're responsible for. You're also responsible for the cricohyaloid ligament. But again, notice nicely, the names tell you everything about it. The thyrohyoid ligament is the ligament between the hyoid bone and the hyoid bone and the thyroid cartilage. The cricothyroid ligament is between the thyroid cartilage and the cricoid cartilage. So again, the names kind of tell you everything about them. However, notice what we don't see in this picture is the mucous membrane that we know lines all of this. They've peeled that away so we can see the underlying cartilage and the underlying bone with the hyoid. But you have to remember that this whole thing is actually lined with our mucous membrane. And that's what you see really nicely in this picture. Notice as you see here, our mucous membrane is intact around the epiglottis and then down, forming our 2 folds, and then continues down into the trachea and our oral passageway. Notice there is this extension, and we'll see another view that'll show it nicely as well. The mucous membrane extends from the epiglottis down to the arytenoid cartilage. And so this is actually called the aryepiglottic fold. With this aryepiglottic fold of the mucous membrane, notice suspended within that aryepiglottic fold is our second paired, or our third really paired cartilage chunk, and that is the cuneiform cartilage. This little chunk of cuneiform cartilage is actually embedded within the mucous membrane, helping to stabilize that mucous membrane and keep it open. And so by doing that, that's why we don't see it in the previous picture. Without the membrane, it would basically just be hovering here in space, right? Hovering here in space, but we don't have that. Instead, we need it in there. Now, when you look at the chart, when you look at the models, one of the things you'll see is as you look at the posterior view, there are 2 bumps, really 4, one on each side, and we'll see these bumps. And again, I think I have a picture that shows this. Ah, that's the picture I want. Wait, go back. Perfect. Excellent. So notice here is the epiglottis. Here is the aryepiglottic fold going from the epiglottis to the arytenoid cartilage. And notice on that, we see these 4 bumps. 2 of these bumps are more medial and they're more inferior. and those are the bumps formed by the corniculate cartilages. And then here, lateral and superior are the two bumps are the cuneiform cartilages. So when we see this from a superior view or when we see this from a posterior view, we will actually see those four bumps in the area epiglottic fold. And so that's where you can actually see those cartilages within there. All right. So as I mentioned, we're going to use muscles, we're going to use ligaments to basically rotate the arytenoid and the corniculate cartilages. When we do that, what happens is what we see here. Here, we see our two folds in the mucous membrane. This first fold is a superior fold and lateral fold. Notice it's well vascularized. This is a thickened part of the mucous membrane. And as I mentioned, this is what is known as the vestibular fold. This is the one that, as we said, has that highly sensitive mucous membrane. So if any irritants come in contact with it, we start coughing violently. Deep inferior to it is a much more rigid, less vascularized membrane or fold, portion of the mucous membrane known as the vocal fold. This is the part that, as we express air, vibrates to produce our sound. And notice when we rotate the arytenoid cartilage, we can rotate it so that those vocal folds open, and now our glottis is open and air can be expulsed. Or we can rotate them closed, closing the glottis so that the air stays in the lungs. So this ability to open and close our glottis is something that, again, you may have experienced this morning for you, because one of the things that we do with this, obviously we open and close it to let air in and out. If you are going to speak like I'm speaking right now, then you need to open that glottis so that the air can come out. But what if instead, right, you wanted to take your morning constitution before lecture? So there you are in the bathroom trying to help to encourage the feces to be expulsed from your body. And one of the ways we can do that is a process we call the Valsalva maneuver. We'll talk about this in more detail when we go back to the digestive system. But basically what we do is we fill our air with lungs, we then close our glottis, and then we contract our diaphragm. Normally that would force the air up and out, but instead, because our glottis is closed, it actually forces the air down. And as we force the air down, that increases the pressure in the abdominal pelvic cavity, and it helps in the expulsion of our feces. So if you ever think about how when you're bearing down when you're trying to defecate, if you're having a more vigorous constitution, then that ability to do that is that Valsalva maneuver, and basically using the air that is trapped in your lungs by closing your glottis to help to increase that pressure and to force the feces out. So again, as we talked about, there are two sets of folds. Oh, sorry, I just never mind. My chat window went crazy. Two folds in the glottis, as I mentioned. The first one is our vestibular. It is superior, lateral, and more inflexible. Right, again, it's got that sensitive membrane to it. And then we have our inferior, medial, and very elastic vocal fold, and that is the one that vibrates. And again, it vibrates like a string of a guitar. So like a string of a guitar, the stronger it vibrates, the bigger the amplitude of the vibration, what happens to your sound?
It deepens.
Well, it gets bigger, right? The larger, right? The harder of a pluck you do of the string, the more the string vibrates and the bigger the sound you get. So you increase the amplitude by getting larger vibrations. However, you were right. If instead I turn the little knob to increase or decrease the tension, then when I pluck the string either vibrates more or it vibrates less. And when it vibrates more or less, what does that change about the sound?
Pitch.
Yeah, the pitch or the tone. The pitch or the tone, absolutely. So if I tighten it, the faster it vibrates, the higher the frequency of the sound. We all do this voluntarily. It is consciously controlled. right? We don't think of it that way because you're not actually consciously thinking about which precise muscle you're contracting to produce a sound, or to change the sound to sound deeper, or change the sound to sound higher, right? You don't think about which muscles you're actually contracting, but it is something that is consciously controlled. And one of the ways we know that is, first, it has to be learned as an infant. It isn't something we're able to do at birth. It's something we have to learn to be able to do to produce sounds and speak. But the other way goes back to our puberty. In puberty, not only does the laryngeal prominence, the Adam's apple, get bigger, but the other thing that happens with all of those androgens coursing through that boy's body is a thickening of the vocal fold. And so as that vocal fold thickens, it vibrates at a different rate. So now the male consciously makes the decision to produce speech and puts tension into that muscle to produce the speech that he normally would. But because of the change of the thickness, the frequency changes. And I think, as we've talked about in class, that person's voice cracks. Right? So you get the cracking of the voice because they have to relearn how to control those muscles. Now, here's the other thing as well. As I'm speaking to you right now into this microphone, assuming this microphone was small enough, and I'll tell you right now it's not, but if it was small enough to shove it in my mouth all the way down to where the glottis is, and I put it in there and we tried to listen, would you be able to understand the words that I'm saying?
No.
The sound I was making would not be a meaningful speech. In fact, all you would really hear is a buzzing sound. Right? So what actually is happening down there is what we are producing, that vibration of the vocal folds, is a process we call phonation. And the result of phonation is just the production of a buzzing noise. Then, again, learned behavior. We use our nasal cavity, we use our oral cavity, our teeth, our lips, our tongue, our mouth to shape that sound into meaningful speech. That ability to take that buzzing sound and convert it into meaningful speech is what we call articulation. And it requires all of those things. Again, obviously, if someone's holding your tongue and you try to speak, you can't necessarily produce meaningful speech that way. You can make some sounds, obviously, but people aren't going to understand you as easily. But you can also call your friend on the phone and instantly know that they have a cold. Why can we instantly know that they have a cold? Because their voice changes, right? When you get that congestion in the nasal cavity, that nasal cavity helps to produce echo. It helps to produce vibrations in that sound. And so when your nasal cavity, is congested with mucus, that changes the tone of the voice. And again, even though they can articulate the words, the sound of it, the timbre of it changes. And so you can tell right away that they have a cold. So again, producing the sound is the phonation. Shaping that sound into meaningful words is articulation. All right, questions on that? All right, excellent. From there, we enter into our windpipe. Again, as it leaves through that one completely circular piece of cartilage, and it is truly the one completely circular piece of cartilage. As we look at this from an anterior view, and I really, really hope I have a posterior view, although I know I have a cross section, we see the trachea. And notice the trachea also appears to have rings of cartilage on it as well. But these rings, like the thyroid cartilage, are also C-shaped. They only go, and I'll do a horrible job of drawing this, they only go 3/4 of the way around. The same way that the thyroid cartilage is open at the back, these tracheal cartilage rings are open in the back as well. And if you think about it, it makes sense that that would be the case. Let me change colors. Because if you think about it, right behind the trachea, as we'll talk about, is your esophagus. Your esophagus is a muscular tube where that cheeseburger that you had for breakfast has to come and pass through. There's the meat, there's the bun, there's the sesame seeds, right? And again, I know everybody in this class takes small bites and chews those bites the 30 times you're supposed to chew food before you swallow it. But there are people who will take a massive chunk out of something, chew it 3 times, and swallow it down. So while our esophagus, being a muscular tube, can expand dramatically to accommodate that food, we don't want that food as it comes down to get stuck against some rigid structure that would block its opening. That could stop the food from continuing down the esophagus. It could also force the narrowing of the airway and cause problems that way. So our airway here in the trachea is going to be open in the back. So again, we have the trachea, and the trachea are going to terminate at the bronchi. Like I said, I'm pretty sure I have a pretty picture. We'll get to that in a second. There's a pretty picture. Now, let me clear my drawings. So here you can see what an amazing job I did of drawing it. Here we have this C-shaped cartilage. Here we have our mucous membrane with our ciliated pseudostratified columnar epithelial tissue and some massive mucous glands. And then posterior to it, we have the esophagus. And here we see it in the illustration, and of course here we see it in our light microscopy. So this is a cross-section view through the trachea, where we can see the esophagus behind it. Now I do want to go back to the previous picture because there's one other thing here that I want to show you. The anatomy of the trachea is pretty much consistent as we work down the entire length of the trachea. However, remember, as we talked about in that vestibular fold, we have a highly sensitive mucous membrane that can cause coughing if any irritant comes to it. At the very base of the trachea, where the trachea terminates and becomes the 2 primary bronchi, this is a region known as the carina. And the carina is another location where we have a highly sensitive mucous membrane. A single speck of dust, right, a single drop of excess moisture can irritate this carina, and it causes a massive, vigorous coughing effect. If you've ever had, water go down the wrong pipe or inhaled like a dust or something like that, and you had that massive, violent coughing attack, that is typically caused by that irritant coming in contact with the carina. This carina is one of the major last lines of defense to make sure that whatever that dust and debris is doesn't get into the airways, doesn't get into the lungs. So any irritant that comes in contact with that carina, right at the base of there, is going to cause that violent coughing to try to cause the expulsion of whatever that irritant is. All right, we have 1 windpipe, 1 trachea, and then of course it is going to branch into 2 primary bronchi. And why would we branch into 2 primary bronchi?
2 lungs.
2 lungs. There you go. Exactly. We have 2 lungs. And so that is where we are going to go next. All right. Excellent. All right. So again, I think this is another good stopping point. Let's go ahead and take our next break here. Take another 5-minute break. We will come back in 5 more minutes. So let's see. My phone says it is 9:31. So let's come back at 9:36. And we will pick up from there. So here, I'll write that down. Back at 9:36 and turn on recording so that I don't forget again. Excellent. So let's go ahead. I'm going to pause the recording again. We'll take a quick 5-minute break and then I'll come back and we will continue from there. Pause. So we have been working our way down to the respiratory passageways, and we are to the bronchi. As was mentioned, there are, let me go ahead and get my text up here, 2 primary bronchi, because we have 2 lungs. And it is these 2 primary bronchi that are going to move towards the lungs, but don't actually enter the lungs. Those 2 primary are going to branch to form our secondary bronchi. And again, quick show of hands. Everybody back? I guess I should have done that before I started talking again. Give me a show of hands or a yes, a thumbs up or something to let me know that everybody's back. Excellent. Perfect. All right. Most of you are there. That's definitely a good thing. Oh, excellent. Perfect. All right. So it is the secondary bronchi that then actually enter the lungs. However, our lungs, as we were talking about, are divided up into different lobes. And there are a total of 5 lobes. There are 3 on the right side and 2 on the left side. That is kind of weird that we have that asymmetry. Anyone have any idea why we might have an asymmetry in the
The heart?
relationship? Yeah, exactly. If you remember when we learned our cardiovascular system, our heart is not. Stephanie, did you have a question, or were you just responding to before?
It was responding to before.
Ah, perfect, excellent. Okay. Yeah. So the heart, remember, is offset to the left side, and so basically there's an indentation, what we call the cardiac notch, in the left lungs to accommodate that offset heart. So there is some asymmetries between the right side and the left side. One of the ways we see that is in the fact that we have 5 total lobes, 3 on the right side and 2 on the other. So not surprisingly, the same way we have 2 primary bronchi, because we have 2 lungs, we have 5, oops, not there, there. 5 secondary bronchi, because it is those 5 secondary bronchi that are associated with our 5 lobes, and it is actually these secondary bronchi that are going to enter into the lobes. So they form outside the lungs, and they're not the ones that actually enter into the lungs. Instead, oops, what's going on? Oh, back to the mouse. There you go. We have that asymmetry. Oh, that's the other important thing. We have that asymmetry in that there are going to then be 5 secondary going to the 5 lobes. And the other big difference that we see is that there's also asymmetry in the shape of our primary bronchi. In our primary bronchi, the right one is typically wider, is typically shorter, and is typically more vertical in its orientation, where the left projects more horizontally. This is significant from a clinical standpoint, because if someone were to accidentally inhale like a pee or something along those lines, then as a result of that, that structure, that substance is more likely to get to fall into and therefore block the primary bronchi on the right side than on the left side. So when there is an obstruction, that obstruction, especially if it's like a food type obstruction, is more likely to occur on the right side than on the left side. For this, I think I am going to finally pull out my iPad. We have not done that yet, so that we can talk about the hilum and the root of the lungs, where we then enter with our secondary bronchi. So let me see, there's the pretty picture that I want from there. But what I want to actually do now is I want to switch to my iPad. So this will take me a moment, a second to get this set up. Perfect. All right. Now, when we look at the ribs, when we look at the lungs from the top, what we see is they have a shape that we are very similar with, familiar with. And that is that it basically is convex on 3 sides. However, on the inside, well, that's weird. I'm holding it. Oh, interesting. Okay. Hold on. I want to start over. I was holding it upside down, and I don't want everything I write to be upside down. So let me do this again this way. So it is convex on 3 sides, and then on the 3rd side, it is concave. All right? This outer region here, all of the convex part, is what is known as the costal surface. Because what, of course, does costal refer to?
The ribs.
Ribs, absolutely. And in fact, if you look at the models, if you look at many of the illustrations, you will actually see indentations in that costal surface where the ribs actually sit up adjacent to the lungs. The concave indented region is the area that is known as the hilum. We've seen this in the lymph nodes. We will see this in the kidneys. We see this type of anatomy in lots and lots of places. Now, one of the important things to remember is that I'm going to change colors here. There's a couple things. First, this hilum is where all the structures attach. To the lungs. That means our blood vessels. That means our nerves. And this is also where our secondary bronchi are going to attach. They're all going to attach into the hilum. And the specific region where these all attach is what is known as the root. Now, there is another thing we need to remember, and I'll switch colors. What color haven't I used? Let's use blue. We know that our lung is associated with a serous membrane on the outer surface, our pleura. And as we know, one layer of the pleura is in direct contact with the outer surface of the lung. And of course, what would that surface be? What would that serous membrane be? How would we identify it?
Visceral pleura.
Yeah, absolutely. This is our visceral pleura. We also know that our thoracic cavity is going to be aligned, and clearly there's not going to be that big of a gap in between them, but I'm emphasizing this for effect. We have the outer one, which of course then is the parietal pleura. But we know one other thing as well. If you remember back to 430, we know that with serous membranes, these two things have to connect to each other. So what actually happens is our visceral pleura basically comes up to where the blood vessels and the nerves and the bronchi are. And it is at that point where it comes off the wall and forms the parietal pleura. So again, the visceral pleura comes down the hilum to where all the structures attach and then comes up to form the parietal pleura. So again, when we talk about the hilum is the entire concave region. So this entire concave region is the hilum. However, the portion of it where just everything attaches and where the visceral pleura becomes the parietal pleura, that specific region of the hilum is what is known as the root. I believe I have a picture that does a nice job of showing this, but I want to make sure to emphasize this because I think this is one of the areas where people do sometimes get confused. There is a difference between the hilum, the entire concave region is the hilum, and then just the part where the blood vessels, the nerves, and the bronchi connect is going to be the root. So if we go back to the lecture, I think if we jump ahead, I think I've got a pretty picture that shows this. There we go, perfect. That's what I want to talk about. So notice now we are looking at, and let me go back to my pointer. So this is that whole entire concave region. This whole entire region is the hilum. And you can see one of the reasons why I'm emphasizing this. Notice they put the label for the hilum right here in the center, which is correct. This is the hilum, but this is also the hilum. And this is also the hilum, and that's also the hilum, and that's also the hilum, and this is the hilum. This entire region is the hilum. The entire concave portion is the hilum. However, notice this part here, and you can see clearly defined by where the visceral pleura is coming and then coming off the wall. This region here where our arteries, our secondary bronchi, and our nerves are all connecting, this area right here and this area only is the root. So this is the root, but the entire area here is the hilum. Can I get a yes or a thumbs up if that makes sense?
Yes.
Got it.
Yes.
Excellent. Perfect. All right. So when we look at the anatomy of the lungs, those are the things that we're looking at. We've got the costal surface, which is the outer surface. We have the hilum. We have the root. I'm going to go back to my drawing because I'm having fun doing that. Okay, not really, because again, my handwriting is so horrible, as you know, but All right, let me start a new piece of paper, and I need to rotate this again. That way. Excellent. The other way, if we think of the lungs are basically cone-shaped structures. They have a nice round base, and then they come up to an apex, right? If we think of their locations, the base sits where in the thoracic cavity? Where does the base sit?
The inferior.
True, inferior, and really, if you think about it, it sits on the diaphragm.
Yeah.
The base of the lungs basically sit on the diaphragm, whereas the apex basically comes up and peaks basically right about where your clavicle, right, where your collarbone is. You feel your collarbone, that's basically where the apex of your lungs come in. So we have those two lungs, the left and the right, that come basically from your clavicle, your collarbone, and then basically sit on the diaphragm. So we have that nice cone-shaped structure. And again, if we go back to the previous picture, so now we can go back to the lecture and, oops, wrong direction, back here, and we can see that here with my spotlight. We can see the base of those cone-shaped lungs is sitting on the diaphragm. And as you can see, the apex basically is coming up here to where the clavicle. We can see his clavicle right here, and we can see where that apex comes up to that. Notice also as we look at this, we see that cardiac notch, that indentation where the heart needs to sit. And we can also see how the two lungs are separate in their divisions. Here on the left side, we have two lobes. We have a superior lobe and an inferior lobe, whereas on the right, we have three, a superior, an inferior, and then a middle lobe. Notice to have lobes, we need big indentations. I remember an indentation or a groove was something we called a sulcus, but remember a large sulcus was a fissure, and that's what we have here. Again, what separates the superior left lobe and the inferior left lobe is the oblique fissure. Notice we have an oblique fissure over here on the right as well, but we also have a horizontal fissure. So the horizontal fissure separates the superior lobe from the middle lobe, and the oblique fissure separates the middle right lobe from the right inferior lobe. So we have three lobes on the right, two on the left, two fissures on the right, one on the left. 5 total lobes, so 5 secondary bronchi. Again, here we see that anatomy. Oops, not what I wanted. Why is that doing that? There we go, and we did that. And then here we see we get rid of the pesky person, and we see the lobes, and we see the fissures really nicely again. Again, these are filling our thoracic cavity, with the exception of the middle region of the thoracic cavity. And what's the middle region of the thoracic cavity called again?
Mediastinum.
Mediastinum, excellent. We have that mediastinum. So again, we did that, we did that, we did that. So perfect, we've looked at all of those things. And we've seen it in all the models, all the illustrations. Here we actually see it in an actual cadaver. So we can see that cardiac notch. We can see the 2 lobes on the left side, the 3 lobes on the right side, and a lot of blackness in there. So this person was probably a good-sized smoker, which isn't surprising. Most of our cadavers are older people in their 80s and 90s, and they grew up when there were cigarette ads on TV, and doctors smoked, and and talked about it on TV about how it calmed their nerves before they saved people's lives, and all the other important things that happened back then as well. All right, we kind of already did this. The lungs are covered by a serous membrane called the pleura. We have the visceral pleura and the parietal pleura. And again, the visceral pleura is on the surface of the organ. The parietal pleura covers the outer surface, really lines the thoracic cavity. And then we have in between our pleural cavity. Now, our pleura is a serous membrane. Let's do some writing. A serous membrane, of course, as we know, is comprised of Oh, interesting. An epithelial tissue on top of a connective tissue. Anyone remember what epithelial tissue lines the surfaces of our serous membranes? Did you have a question, Imra?
Simple cuboidal.
Close. Remember, we want a barrier. We want it to be as thin as it possibly can be. So it's actually a simple squamous epithelial tissue. And of course, what connective tissue is it?
Areolar.
Areolar. Now, this areolar connective tissue, is it a lamina propria? No, remember, areolar connective tissues are just lamina proprias if they are part of a mucous membrane. However, if you remember when we talked about the heart, serous membranes, they're simple squamous epithelial tissues based on their location. Remember, we call them a mesothelium. So we do have that mesothelium of the simple squamous epithelial tissue. The connective tissue is an areolar connective tissue. And of course, the function of a serous membrane is to do what? What do serous membranes do?
Reduce friction.
How? Absolutely correct.
Secreting fluid.
Yeah, absolutely. They produce serous fluid. Now, again, is our goal to fill that pleural cavity with as much serous fluid as we possibly can?
No.
No, we just want a thin layer of it, right? And again, remember that thin layer of serous fluid, remember, based on its location, when we talked about this in the cardiac system, in the heart, we call a transudate. Again, we're still not getting away from that pesky tissue stuff that we learned in 430. So again, based on its location, we call that a transudate. So there we go. Excellent. So that's our job. We produce that thin layer of serous fluid, lining those surfaces. And as was mentioned, really, and here it's really important, it does two things. One of the things it absolutely does is that it reduces friction. Oops. Right? Between the lungs and the thoracic cavity. However, remember the other thing that we talked about when you have that glass with condensation on that glass countertop, the other thing we want is here we actually want surface tension. Right? If you think about it, we don't actually have any muscles in the lung itself. Right? What we do is we contract muscles to change the thoracic cavity. We change the shape of the thoracic cavity. We contract muscles like the diaphragm, our sternocleidomastoid, all of those things to move our thoracic cavity. And as our thoracic cavity moves, we want our lungs to move along with it. And so basically we want surface tension so that when the ribs pull away, that surface tension is going to keep the lungs attached to the ribs so that the lungs will expand when we change that shape of our thoracic cavity. So here in this case, the other thing that serous fluid is doing is producing that surface tension because of the water there. So we want that surface tension, we want that water, we want that transudate so that it can reduce friction, but also keep the two anchored to each other. And again, we see all of this, whoops, wrong button. Back there. We want that pleural fluid in there to, again, produce that surface tension and to reduce friction. And here's the pretty picture. So now I got to clear my drawing. Where again, here we see the visceral pleura on the surface of the lungs. Here we see the parietal pleura lining the cavity. Notice here again, we have the hilum on this side, but notice where the visceral pleura comes off to form the parietal, visceral pleura comes off to form the parietal. This region right here is our root. So again, here we see that root. All right. And so again, it is the primary bronchi that come towards it, but then it branches to the secondary, and those secondary go to the lobes. And how many lobes do we have again?
5.
And so how many secondary bronchi do we have again?
5.
There you go. Perfect. All right. So there you go. And look, I got the pretty pictures that show all that too. Excellent. All right. Questions on that? All right. Excellent. So again, one of the things we've been talking about, and I'll write it all out again, our primary bronchi. We have 2 primary bronchi because we have 2 lungs. That makes sense. We have 5 secondary bronchi because we have 5 lobes. So that's not new. We've already talked about and made sense of that. But we also have tertiary bronchi. And again, we have a meaningful number of them. As it turns out, and remember, not only do we have asymmetry from individual to, from right to left, but there's also some variation from individual to individual as well. So we can have anywhere between, let's say 16 and 20 tertiary bronchi. The reason for this, again, is meaningful because our lobes of our lungs are, again, the same way we have 2 lobes, the same way we have 2 lungs that are meaningfully divided into 5 lobes. Those 5 lobes are meaningfully divided as well. They are meaningfully divided into what are actually known as bronchopulmonary segments. And if we have 16 to 20 tertiary bronchi, guess how many bronchopulmonary segments we have?
16 to 20.
16 to 20, exactly. A bronchopulmonary segment is basically a section of our lobe, a section of our lung, that basically has 1 tertiary bronchi and 1 pulmonary artery and 1 pulmonary vein that all feed into it. So it is a compartment that is separate from the other compartments. Each of these is a separate independent compartment. Our lung is not just a loose random collection of alveoli like a big cluster of grapes. And even a cluster of grapes isn't truly just a cluster of grapes. If you've ever really inspected a cluster of grapes, there's a main stem, and then there are separate stems that come off of it. And so a separate stem that comes off has its own grapes on it. And it's the same thing here. These bronchopulmonary segments are meaningful, not only from a functional standpoint, where 1 blood vessel feeds in, 1 blood vessel feeds out, and 1 airway feeds in, but from a clinical standpoint as well. Typically, when cancers occur or pneumonias occur or things along those lines, typically they just occur first in 1 bronchopulmonary segment. So if a cancer, for instance, is caught early, they can basically go in and excise that bronchopulmonary segment by cutting 1 blood vessel, 1 artery, 1 vein, and 1 bronchi. We can basically pull out that segment and hopefully save the rest of the lung. So we don't just have to remove the elevate lung. Now, again, of course, the longer the cancer stays in there, the more it's going to spread to other areas. But it is clinically significant that way. And I believe I have a pretty picture that shows this. So again, I'll make sure everybody gets this point. Again, there's a meaningful number of primary bronchi, 2 primary bronchi because of 2 lungs, 5 secondary bronchi because of 5 lobes, and 16 to 20 tertiary bronchi because there are 16 to 20 bronchopulmonary segments. So again, our primary bronchi form the secondary, 5 of those. Our secondary branch form our tertiary, and those tertiary supply our bronchopulmonary segments. As I said, there's asymmetry in the lungs and asymmetry in individuals as well. I love this picture from your textbook. Basically, what they've done with this picture is that they have color coordinated all of the tertiary bronchi and other the further branches that come off of that. Primary bronchi that tertiary bronchi come off and go to it. So basically what we see in these color-coded areas, this brown one, this pink one, that blue one back there, that green one, this lighter blue, the red one, all of these color-coordinated points correspond to the airway parts of a bronchopulmonary segment. Now remember, blood vessels are going to these things as well. But you can see each one is its own compartment. Now, not surprisingly, there are differences between the right and the left because of that cardiac notch. Everybody in this classroom, everybody in the world on their right side all have 10 bronchopulmonary segments. 3 in the superior, 2 in the middle, and 5 in the inferior lobes. And that is true for everyone. However, over here on the left is where there's variation. Everybody can be different. Some people may have 10 bronchopulmonary segments on the left. Some people can have as few as 6. Now, as far as we can tell, this is just more of a plumbing issue. There doesn't seem to be any difference in function or any difference in a predisposition for any kind of respiratory diseases or anything else those lines. It's really just difference in the way the pipes were laid in these. So again, that difference isn't meaningful or significant, but there is a difference. So this particular lung, notice that we're looking at, has 9, 4 in the superior and 5 in the inferior, but that can vary dramatically. So the numbers over here on the left side aren't meaningful because it can be different from person to person. But what this does show is 2 things. One, it shows our tertiary and how they're meaningful because they go to these bronchopulmonary segments. But as you also see, there can be plenty of other branching that is taking place as well. And in fact, as it says on the previous slide, as you can see, after the tertiary branching, there can be as many as 21 orders of branching that continues to occur. So that means there's going to be quaternary and pensionary and so on and so forth through all of the different branches. However, the other branches aren't going to be meaningful in the same way that the first, the second, and the third. The primary bronchi, secondary bronchi, and tertiary bronchi, we have a set number because they're meaningful in their branches. The rest of the ordering don't branch to as many meaningful regions the same way. Eventually, they'll end up individual lobules, but it isn't as significant that way. Lastly, when we get down to the smallest bronchi, and when we talk about the anatomy of the bronchi, we'll see what the difference of those are. The smallest bronchi are what are known as bronchioles. And the bronchioles are the last portion of our conducting zone and in our respiratory passageway. All right, questions on that? All right, let's take a look at our bronchi, and we can see here we see our bronchial tree, and it really does look like a tree, especially if you take it and look at it upside down. Notice, as we mentioned, here's our single primary bronchi. Well, I guess there's two primary bronchi. This guy, we're just going to the left. That one primary bronchi is going to feed into the 2 secondary bronchi. And those 2 secondary bronchi will then divide into however many, somewhere between 6 and 10 tertiary bronchi. And then from that, the bronchi just get smaller and smaller. Now, obviously, the difference in size is one of the ways that these are different. But there are a couple other very meaningful ways that our bronchi are different. So when we talk about differences in the bronchi, obviously the biggest difference is size. They're going to get smaller as they go. However, another big difference in our bronchi, as you can see from just looking at it superficially, is the shape of the cartilage. So notice in the primary bronchi, and let's do this here, in the and I'll just put PB for primary bronchi. Notice we still have those C-shaped ring cartilages like we have in the trachea. So again, remember, this is a C-shaped cartilage that doesn't go all the way around. We have the same thing in our primary bronchi as well. So it's a C-shaped ring of cartilage. Oops. Ring of cartilage. You can see I type as well as I spell on the board. However, notice as we move on to our secondary and tertiary, as we go from the secondary and below, notice what happens is instead of rings, we start to get irregular shaped plates. Of cartilage. Those irregular shaped plates of cartilage are going to still allow the airways to stay open. But the big important significance of this is that it doesn't hold it into as rigid as a structure. Here the trachea, here the bronchi, that big C-shaped cartilaginous ring keeps it open, and it's also very solid. However, as we know from talking about the sympathetic and parasympathetic nervous systems, we want to be able to dilate the airways, increase the amount of air that comes in, or constrict the airways, decrease the amount of air that comes in. So here in these smaller bronchi, we typically have more smooth muscle. And that increase in smooth muscle, along with the change into the irregular plates of cartilage, allow this region to expand and contract more so that we can let more air in or less air in. So when you take that EpiPen with that anaphylactic shock, these are the regions that are dilating. The smooth muscle relaxes, those airways open up, and those plates allow for more expansion and contraction of the tissue in that area. Notice, and I'm cheat and come back to my highlighter here, as we come down towards the terminal end of this, I wonder if I can continue to write on the one. Nope, I have to start new. Notice at the end of this, eventually, the cartilage goes away. And as you can see, where the cartilage goes away, there's no more cartilage in those areas. That where it goes away, those are the airways that we call the bronchioles. So while a bronchiole is obviously a small bronchi, it's more meaningful than that. In the same way that an arteriole is a small artery. But remember, an arteriole did not have a fully complete all three tunics the same way that an artery does. And it's the same way here. A bronchiole is a much smaller bronchi, but it's also in some ways an incomplete. It doesn't have, for instance, the cartilage. So one of the big changes is the change in size. One of the big changes is the change of the shape of the cartilage. But there is a third big difference. And that third big difference between the bronchi is in the epithelial tissue that lines them. Again, for our primary bronchi, as we know, it is that ciliated, pseudostratified, columnar epithelial tissue. However, as we start going from the secondary and below, it transitions into simple columnar. And by the time it reaches the bronchioles, it becomes, oops, simple cuboidal. So as we transition in our bronchi from primary all the way down to bronchioles, there are three big changes we see. We see a change in size from bigger to smaller. We see a change in the cartilage from cartilaginous rings to plates, irregular plates to none. And we see a change on the inside in the epithelial tissue from ciliated pseudostratified to simple columnar and eventually to simple cuboidal. All right. So again, if we, I think I have to clear the board to be able to do this. Give you guys a second to, again, make sure you get that down. By quick yes or no, you guys have a little yes or no things. Give me a yes if you're taking notes, or a no if you're just watching and observing. Oh, cool. Wow, awesome. All right, excellent. It works both ways. So again, that's cool. Again, the nice thing to remember is we will be posting this, so you do have the opportunity to go through it again and make the notes that time. But that's cool. Okay, I will try to keep that in mind. I especially in the first hour, I think hopefully I've gotten a little bit better. And again, hopefully you guys will give me feedback on this. I think especially in our first hour or first 45 minutes or whatever it was, I felt like I was talking really fast without getting to interact with you guys in quite the same way. I think my tendency is to hit the gas with my talking, and so I will try to be cognizant of that and do my best to talk slow enough to give you guys time to take these notes and things like that. And hopefully me taking the time to write things on the screen will help with that as well. All right, so like we said, our epithelium mimics the trachea at first with that ciliated sort of stratified, but as the bronchi become smaller, there are three big changes. Obviously the size, but we also see again the change in the plates, change in the epithelial tissue, and as I mentioned also, I guess there's really a fourth change, as we talked about, more smooth muscle, which makes sense. If this is a cartilaginous ring, having smooth muscle in it, there's only so much, that's something I have to go back and do. There's only so much that movement that we can have of this structure, whereas obviously with the plates, it can expand and contract a lot more. That doesn't mean, and so actually now I get to go all the way back in my lecture, way back to when we talked about the trachea. That doesn't mean, while notice within the wall of the trachea, there isn't a lot of smooth muscle. Doesn't mean that there isn't any smooth muscle and that we don't have any ability to change the shape. Notice, and again, it's not even labeled on the illustration, but it is on the light microscopy. I just think it's easier to see here. Notice at the base of the C-shaped rings, there is a small, thin muscle. This is a smooth muscle. And this smooth muscle is what is known as the trachealis. The trachealis does help us to modify the size of the lumen of the trachea. When this contracts, it gets more narrow. When it relaxes, it enlarges. So there is a little bit of smooth muscle, but notice it's not within the wall of it so much. There is a special smooth muscle structure here known as the trachealis. And so that trachealis is the, perfect, there you go, exactly right there. That trachealis is smooth muscle. So we do have some smooth muscle, but notice it's not in the wall of the trachea. And the primary bronchi are the same way. So the primary bronchi are the same way, but by the time we get down to the part where there are irregular plates, there we go, oops, too far. Then in here, there is more smooth muscle. So down here, there is going to be more smooth muscle there. All righty. Questions on that? All right, excellent. As I mentioned, notice everything we've been talking about so far, from the nasal cavity and all the way down to these bronchioles. These have just been part of that conducting zone, whose sole purpose is to move and process the air. Right? We want to filter it, we want to moisten it, we want to warm it, until we get down to the very end. What we see here is our very last part of the conducting zone, right? Which is why it's the terminal bronchiole. Notice there's no cartilage, right? We have our smooth muscle, we have our epithelial tissue on the inside, which at this point is going to be hold on one second. Sorry. Our simple cuboidal epithelial tissue. So simple cuboidal epithelial tissue here, smooth muscle, no cartilage. These are bronchioles. And this last one that is just part of the conducting zone is a terminal bronchiole. Notice this terminal bronchiole feeds into another bronchiole. No cartilage, smooth muscle, lined with a simple cuboidal epithelial tissue on it. However, this bronchiole has a ball-shaped alveolus on it. This alveolus is like the alveoli that we see down in here. And if you have an alveolus, that alveolus is where gas exchange is going to take place. So here we have a bronchi that has alveoli. Here we have a bronchi that doesn't. The bronchi that doesn't cannot participate in gas exchange, so it is a terminal bronchiole. However, this one that does have alveoli can participate in gas exchange. Now again, this bronchiole has two alveoli on them, right? So obviously it's not a major site of gas exchange. But technically, if you can participate in gas exchange, even if it's a small amount, you are considered part of the conducting zone. And so the terminal bronchiole is the end of the conducting zone. This is the beginning of our respiratory zone. Our respiratory zone is where this respiratory bronchiole begins, and this is where gas exchange can occur. So again, conducting zone just conducts. Respiratory zone is where we get our gas exchange. And so this is that break. This is where we go from conducting zone with no alveoli, no gas exchange, to a bronchiole that has alveoli, so can, even if it's just a tiny bit, it still has the ability to participate in gas exchange. And since it can participate in gas exchange, it is part of the respiratory zone. Now again, this respiratory bronchiole is going to feed the air into our major alveoli, and this is where most of the gas exchange is going to take place. But even if you just have one alveolus, right, you get to help participate. And so you're considered part of the respiratory zone. All right, let's take a look at that respiratory zone. Here we see it in the pretty picture. Here we see this great illustration. It's not really an illustration, this great light microscopy of showing the anatomy of this. So we see this here. Again, the majority of our respiratory zone are the alveoli. Again, I'm not the one who counted it, but I'll take their word for it. There are over 3 million alveoli per lung. In the back of our classroom, we actually have a lung that has been basically freeze-dried. And if you've ever felt it or get the opportunity to feel it, it basically has the density and the weight of basically just styrofoam. It's like this big, huge styrofoam, light, fluffy thing, because as we see here, the majority of it is just these air-filled spaces. Alveoli, as we talked about, are a simple squamous epithelial tissue so that a gas exchange can take place. Now, I'm going to go back to the previous picture because there's some more anatomy we need to talk about. Notice our respiratory bronchiole feeds into a structure known as the alveolar duct. I think of the alveolar duct as the hallway. If you've ever been in a hotel and you go up to the 16th floor where your room is, when the elevator door opens, you have this big, huge, massive hallway. And really, the only purpose of that hallway is to connect the rooms together. So really, basically, you have a ton of rooms that the doors open up into a communal space. And that communal space is where the doors all connect. And that's essentially what the alveolar duct is. In fact, when we look at this histologically, the way you find the alveolar duct is the lack of space. So notice here, we have a respiratory bronchiole. If we had a higher magnification view of this, one of the things that we would be able to see, I'm going to switch the arrow, see if that like that. This is lined with a simple columnar epithelial tissue. We can also see some of the smooth muscle in it, so that's why this is a thicker structure. We can tell this is a bronchiole. Notice we can tell it's a respiratory bronchiole because of this right here. This right here is an alveolus. So what I have is a simple simple cuboidal structure, the bronchiole, with smooth muscle in it that can participate in gas exchange because it has an alveolus. So the fact that it has an alveolus means it is a respiratory bronchiole. It feeds into this void. This void that we see is that space. This is the hallway of the hotel that all the rooms connect to. So notice all of these are the openings to the alveoli, the openings to the room. So really that hallway is nothing by itself. It's just the space that connects all the rooms together. And so it makes it very easy to find the alveolar ducts on the histology slide. And again, when we look at alveoli like on a model, again, they thankfully cheated for us here by cutting it open. But normally when you look at the top, all you see are the grapes. It really is like a cluster of grapes. You go into your fridge, hopefully you've hoarded some grapes. You grab that cluster of grapes out and all you see are the grapes. But you know it's not hope holding those grapes together. You know that if you look inside, there is a stem that all those grapes connect to. And that's basically what the alveolar duct is. The alveolar duct is the stem or the hallway that holds all of those alveoli together. So we have those alveoli accounting for the volume of the lungs. It has a massive surface area. Again, somewhere on the order of 70 to 140 meters squared, which, like we said, is kind of close to the surface of a tennis court. And that's what's sitting inside of your chest, all folded up in on itself. Oh, didn't know I could do that. That's cool. All right. Oops. There we go. Here, we've pulled them out of the lungs so we see a little bit better. Again, notice we have our terminal bronchiole with no alveoli on it, our respiratory bronchiole that have alveoli on it, and then they feed into the alveolar duct, that space that holds the alveoli together, forming a structure we call the alveolar sac. So the respiratory bronchiole feed into the alveolar duct, which feeds into the alveoli sac, which is a collection of alveoli. And of course, if we were to just look at one, the singular is alveolus. Perfect, we did all of that. We're doing good on time. Excellent. All right. The last little bit of anatomy, as I promised, we do need a little bit of microscopic anatomy to talk about. And what we can see here is our anatomy. Here we see our alveoli in an electron microscope. They're the ball-shaped structures. And notice there are all these capillaries that twist and wind around them as well. If we take a cross-section through it, what we get is a pretty picture that looks like this. Now, let me grab my iPad again so we can draw one of these for ourselves. There we go next. Yeah, perfect. Excellent. So what we're going to talk about now are the cells and the structure of a single alveolus. So let me switch my view back to the iPad. And let's get rid of my lungs. All right, excellent. As we saw, an alveolus is basically a ball-shaped structure. Here, and only here, is where we have our simple squamous epithelial cells. The simple squamous epithelial cells that make up the alveolus. So again, they are the wall of the alveolus, the primary structure of the alveolus, are what are known as the type 1 pneumocytes. Pneumocytes are the cells of our alveolus. And if there is a type 1, you can guess that there are 2 types. So they make up the ball-shaped structure. All right. Now, and we'll go back to the picture and look at this in the pretty picture again in a little bit, but I'm going to go ahead and do my own drawing of this here. Oh, did that do that? That works. Okay. Oh, actually, I'm going to cheat because I think it's green on your model. They are the walls. However, what you will see sometimes embedded in the walls are some additional cells. These cells are within the walls, so they're forming part of the structure of the cell, of the alveolus. But these are what we call our type 2 pneumocytes, or what are also known as septal cells. Septal cells' job is to produce and release a substance called surfactant. Surfactant basically, its job is to break up surface tension. Remember, surface tension is that special characteristic of water. The hydrogen bonds of water give it a surface, so that's why when you put a drop of it on the table, it doesn't just spread out all over the place, it forms a droplet, right? Surface tension is what allows light insects to move along the surface. Surface tension is what makes it hurt when you do a belly flop off the high dive. That's the surface tension of water. And surface tension is also, as we talked about, what holds that glass of water to the glass countertop outside on the patio. Water has surface tension, and so it attracts itself. When air leaves our alveolus sac, we don't want our alveolus sac to collapse. Remember when we talked about when we blow up our balloons, we don't want to have to use a tremendous amount of force to get the 2 sides of that balloon to separate from each other. So instead we stretch it out. We break up the tension, we break up the connection so it's easier to fill up. In this case, if the air leaves, the water, because remember there's a massive amount of moisture inside of these so that the gas exchange can take place, the moisture from the 2 sides could cause them to be attracted to each other and cause the alveolus to collapse, right? So it would collapse in on itself, and then it would be almost impossible to inflate it again and get air in there. We don't want that to happen, and so that's what our septal cells do. Our septal cells produce that surfactant, which breaks up the surface air tension and allows the airway to stay open, right? Surface tension is an excellent thing, an important thing on the outside of the lungs, but on the inside of the lungs, it's a bad thing. And so we have the surfactant to get rid of it. Now I need another color. There is an additional cell. This additional cell is not actually part of the wall of the cell, but it is a cell that can move through the walls, can move from cell to cell. And essentially what it is it's a white blood cell. Oops. This white blood cell is a macrophage, or what are known as our dust cells. So if we go back to our pretty picture from the textbook, and I will go back to my pointer. All right, so notice here, gotta stop that. Here we have our type 1, so the beige cells here forming the wall here. These are our type 1 pneumocytes. They're just the simple squamous epithelial tissue that are forming the wall. But notice embedded within the wall are these green cells. These green cells are the type 2 pneumocytes, or what are also known as septal cells. And their job is to produce that surfactant. That surfactant is basically what breaks up the surface tension in this and keeps it from collapsing in on itself. And then remember, as I also mentioned here, the blue ones. Now again, these blue ones are not actually a part of the wall. They can migrate from cell to cell, or from through the cells. They can migrate from alveolus to alveolus as they move through. These are our phagocytes. These are our dust cells. And their job is to absorb any pathogens that got down here. They actually help to play a role in regulating the water content so that there's not too much fluid in here, right? And things along those lines. So this is part of our protection. Some of our second line of defense, or really first line of defense at this point, because this is still outside of our body to help to keep things from harmful from getting in. Notice one more important thing. We're going to talk a lot about gas exchange when we get to this. Notice here is this big alveolus, and here are the capillaries. Remember, capillaries are tiny, so small that the red blood cells basically have to line up side by side, single file, to work their way through the capillary. And so there are often many capillaries around a single large alveolus, and the alveoli are much, much larger than the blood vessels are. We'll see why this is important in gas exchange, because when it comes to gas exchange, right, normally like diffusive processes, right, if we, I'll cheat and go back to my illustration. Because again, this is another point that it is important for us to make sure we understand. So let's go to the next page. And I need to rotate that one as well. Excellent. So I'm up here. Perfect. Okay. If you think back to 430, when we were doing diffusion, right, we had 50% of sucrose on one side and 10% of sucrose on the other. And we know things move from a high concentration to a low concentration until we reach equilibrium. And of course, equilibrium would be 30% on both sides. All right. However, assuming we were in the classroom, which of course we are not, but assuming we were in the classroom and you had your can of soda pop for your breakfast and you were able to open in the classroom, which of course you are not allowed to do. But if you did open in the classroom, that soda pop may be, and I'll just throw a number out here because it's completely arbitrary and I'm making it up. Let's say it was 30% carbon dioxide inside of that soda can. Anyone know what the amount of carbon dioxide in the classroom is? It's okay if you don't, we haven't talked about it yet, but I'll give you a simple answer. It is less than 1%. So the carbon dioxide in the room that you're in breathing right now is less than 1%. Wait, is that greater than? Did I do that wrong? Is that right or wrong? Thumbs up, thumbs down. That's greater than, isn't it? Yes or no? Did I do that right?
I think you're right.
All right. Here, I'm going to cheat, just write less, because my brain's not working for that anymore. All right, less than 1%. So does that mean when I open a can of soda, the percent of carbon dioxide in the room is suddenly going to become 15% carbon dioxide?
No.
No, absolutely not. So the reason for that is because the volume of carbon dioxide that is in that can of soda versus the volume of air that is in the room is much, much different. And so it's going to be the same way when we start talking numbers. And again, numbers are going to be important on this. When you open that can of soda in the room, what happens is you reach equilibrium. But equilibrium is basically when the carbon dioxide leaves the can into the room, not changing the room. So essentially what happens is the gas in the can becomes equal to the room. So when you open that can of soda and you leave it on the counter for half an hour, what happens?
The bubbles are gone. It starts to flatten.
It goes flat. And the reason it goes flat is all the carbon dioxide comes out of it. And that's what happens here. There's so much more gas in the alveolus that the partial pressures of the gas in the alveolus are going to cause the changes in the capillary. So it is right here between this big, huge, massive alveolus and this tiny little capillary where our gas exchange is going to take place. All right. So it is here where we have our air-blood barrier. It is here where we have, again, the alveolus wall, which is a simple squamous epithelial tissue, our pulmonary capillary, which of course is a simple squamous epithelial tissue, and basically the the basement membrane that holds them together. So here we see our respiratory membrane. Our respiratory membrane is comprised of the epithelium of the alveolus, the endothelium of the capillary, and the basement membranes, or again, basal lamina, diffuse basal lamina of this. Again, there's not a smaller tissue in the body than a simple squamous. So when we put two simple squamous epithelial tissues together, and just the glue, the basement membrane holding them together, this makes this an incredibly short distance that our gases have to travel. In effect, the distance from the capillary to the alveolus or back is less than half of a micron. So it's a very short distance these gases have to travel, which again, because we are dealing with diffusion, we want huge surface area, we want short distances to maximize the efficiency. All right, questions on that? All righty, excellent. The good news, that is everything that I had planned for our first lecture. There's a teeny bit more anatomy we need to talk about, but that takes more exploration and more story. So I think that will be best saved for the next lecture. So what I'll do is I will keep the recording going. If anybody has any questions, if there's any part that I went through too quickly and you would like me to reiterate or go over again, or if there's any questions on any of the material we've covered or any questions on anything else, I'll stick around for at least the next 15 minutes. Or until if you go beyond 15 minutes of questions, and certainly I will keep going. But I will stay at least for 15 minutes, so that if anybody has any questions or any additional information they need, I'll happily provide that. Otherwise, I will then save this. Based on the practice one we did, it can take as long as an hour for it to both between me converting it from the Zoom, because that takes some time, and then uploading it to YouTube took a tremendous amount of time for their processing of it as well. So my guess is that sometime within the next hour or so, this will be posted on my YouTube channel so that you have the opportunity to go back to it again. So thank you guys very much. Like I said, I'm happy to answer any questions. Hopefully this format is something that will work for us. Again, I appreciate I went a little fast at the beginning, but hopefully we can work out those things. And if you have any other thoughts or suggestions to help this to be more successful, I would greatly appreciate it. This is the very first real lecture I've ever done online, so this is new for me as well. So I would appreciate any advice that you guys can provide to help this to be a better learning experience for you. All right, so thank you very much, and now the lab is yours. Any questions? And again, remember, you can type them or you can speak them. I'm going to stop sharing my page. Excellent. Oh, no, I didn't. What just happened? Oh, I paused it. I didn't mean to do that. This is what I want to do. I want to stop sharing. How do I do that? I don't actually know how to stop sharing. Oh, there it is. There, I figured it out. All right, perfect. All righty. So again, any questions or anything, feel free to raise your hand or to ask, and I will happily answer those. Otherwise, thank you guys very much and have a good day. So one question that I just got real quickly. Again, remember, if we do have a lab exam, well, let me rephrase that. When we have our lab exam, it will hopefully be in person in class, but if not, it will be online. So you do need to be studying your anatomy list. Again, just like we did with the cardiovascular, My goal is to cover a lot of it during the lecture as we go through it. I'll try to be cognizant of that and try to do a good job of that, especially because of this format. But there is the expectation. Oh, that's what I wanted to show you. There is that expectation that you are going to be working on this material and learning this material on your own. So the other thing that I wanted to show you, so can I share? Perfect. So this is that link that is on our Canvas page for the Cosumnes River College's Virtual Anatomy Lab. So notice as we go through here, you go to the respiratory system. Notice not only do they have histology, but you can click on pictures. So here's that infamous half head chart that we see all of the time. Notice you can see it with labels and without labels. So you can challenge yourself with the material that way. Oh, I love this picture. Oh, this is awesome. I'm going to have this up during the lecture next time. Notice here on the posterior view, can you guys see my mouse?
Yes.
Okay, excellent. Because I can't see the chat from this page right now. So, oh wait, maybe I can bring up. Can I bring up the chat? Okay, yeah, perfect. I can at least see the chat. Perfect. I can't see the participant thing though from this. So notice here from this posterior view, this is the epiglottis. This is the area epiglottic fold. And these are those bumps that we were talking about. These two are the cuneiforms that are noticed more lateral and more superior. And these are the corniculates. So hopefully those as we work our way through that. So there you go, perfect. Cuneiform cartilage, corniculate cartilage, epiglottis. Notice they don't have the area epiglottic fold labeled on this. Notice not only do they show us the nasal septum for this view, but we also see the internal nares as well. So this is the internal nares that feeds into the pharynx. Notice this is a posterior view. Basically what they've done is cut open the pharynx so that we can see all of the structures. That's the oral cavity. So this would be the oral pharynx. This is the nasal pharynx with the internal nares and the septum. And then this would be where the laryngopharynx is, where this is here. So we have that. So we have all of the charts. We have some models. Notice this one's great because it shows the olfactory nerves coming through the cribriform plate. So you can see a lot of these things. Here's that thyroid model. So here we see that arytenoid cartilage and how it sits on top and all those things. Here we see the thyroid. So notice there are a lot of really great models with tags. This is going to be on the exam, I guarantee it. This model is wrong from this up, but we'll talk about that later when we get to that. But so this will be awesome to have this when we get here next week. From here, this is a perfect view. Awesome. So there's a lot of great materials on here. So if you look, we have all of that. So great pictures of all the models. These are all models that are in our classroom as well. So it's a great way to study those things. And if we go back, come on all the way. There we go. Notice there was also some histology as well. So here we see some histology. Looks like this is a cross section through our trachea. We can see the cartilage of the rings there. So there, and notice again, labels that esophagus, trachea. So there it is as well. Ciliated pseudostratified columnar, mucus glands, the hyaline cartilage, all of that stuff as well. Oh, great cilia on this. So there's some really cool pictures on here. I haven't even looked through all of this. So this is a great resource as you are studying this material and wanting that lab opportunity. While we obviously don't have the time in the classroom, what this virtual anatomy lab can provide is it can provide you those resources. And it'll have the same thing for the urinary system and the reproductive system and all of those. So this is an excellent, excellent way to study this material. So since you don't have the time in the classroom resources are here. And we talked about recreating this, but since they've done such an amazing job, it's silly for us to recreate it. So I've made that link available so that you guys can use that. So hopefully this is something that can help you to be successful. All right. So yes, so you're absolutely either online or in person. We are going to have a lab exam. So please do not forget to study the anatomy. I will try to cover most of it like we did, like for instance, the cardiovascular system. But in the cardiovascular system, I was not able to, during lecture, talk about every single item on that list. But that doesn't mean that you're not responsible for that list on the exam, right? So I don't think we ever got a chance to talk about the ligamentum arteriosum when we were talking about the heart, but I know that was one of the things you needed to know on the lab exam. So it's going to be the same kind of thing here. So times like this would be the time to ask questions if there's anything on that list you're not sure about, and that resource. And like I said, if you find more, let me know. If I find more, I'll post them on that same module inside of Canvas to help you guys to get the lab portion as much as possible. And then the PhysioX will help as well, because then we get the physiology a little bit that way as well. All right, excellent. So for the exam, if we do it online, it'll probably slightly different. Again, what I did for the 430 class, the lab exam is pretty much the same. One question, one picture, right? And so you go through the material that way. So is this the PowerPoint presentation, which is kind of like what we did for the endocrine and lymphatic. So it'd be that kind of format, just be kind of doing it online, one question at a time that way. What I did for the lecture exam, and here's the other thing too, again, I am still going with the hope that this is a transient situation. I know there are some rumblings that this could go longer and we may have to do the entire semester online. I'm really, really hopeful that will not be the case. But I don't know yet. So my hope is that's not the case. But one of the things I want to do, because I don't like this format as much, and especially because it was such a quick turnaround, if we are required to do all of the remaining labs, remaining exams online, then I will be taking full advantage of the Proctorio system, where you have to have a webcam and you have to record your surroundings and all the other things that go along with that. The students in 430, I was worried wouldn't have time to get all that set up. So while there are restrictions, I locked down their computers and did things along those lines. The cameras were not required because not everybody had them and had them set up yet. And so one of the things I did is I de-emphasized the exam. And the way I de-emphasized the exam was by making it for fewer points. So for instance, one of the things that they didn't have, there were no multiple choice questions on theirs. Because a lot of that's just, it's not filler. It's not that it's not important, but it allows you to cover a lot of material in a smaller amount of time for something like that. But what I focused on instead were essay questions. However, if we have to do it and I'm going to have to proctor it properly, then yeah, my guess is that it would be the similar type of thing. Multiple choice questions, although my guess there would be fewer of them. And then, short answer and essay questions. So yes, all of that would be there. The 430 did not have any multiple choice questions, but they had essay questions. They had short answer questions. They had a lab exam. And so you will at least have that. And like I said, if we have to go full board, then there'll probably be some multiple choice questions as well. But like I said, I have my fingers and toes crossed that we'll be back after spring break. So our exam would be the week after we return from spring break. So we would have Monday lab and class, and then Wednesday would be the exam. That's the new schedule, the way it's set up. And I'm hopeful that that will be the case. Nasolacrimal ducts. Great question. Let's go back here, because I think we may have, I'm hoping the model that had it will have it. And, that was that it? Yes, perfect, excellent. Okay, so nasolacrimal duct, as the name indicates, is the passageway from the orbit of the eye to the nasal cavity. You are constantly producing tears. They start in the lacrimal gland, which is the superior lateral portion of your orbit of the eye. Those tears wash across the surface of your eye and drain in the medial inferior part through that hole in the lacrimal bone, which is where our lacrimal duct is located. As we talked about in 430, that nasolacrimal duct empties into the nasal cavity so that the tears mix with the mucus, again, helping to moisten our air that comes in. But as we also talked about, if you start overproducing tears when you start crying, because you have to lecture online and your tears are streaming down your face, you also have an increase in tears entering that nasolacrimal duct, entering into the nasal cavity, overwhelming the cilia. And so not only are tears streaming down your face, but snot's running out your nose as well. Notice here, this particular model, we have our superior nasal concha, we have our middle nasal concha, and we have our inferior nasal concha. But notice they've cut a notch out of the inferior nasal concha because lateral to the inferior nasal concha in the wall, that little opening we see right there, that is where your tears drain from your eye. So from the eye, in this case, that would be the right eye tears drain through the lacrimal bone down through the lacrimal duct into this little opening, which opens into the nasal cavity. And so this is the nasolacrimal duct. This is the opening there. We see this little notch in the inferior nasal concha. So you can see their lateral there is where the tears would come in. So that is where that is located.
We can't see the screen.
You can't see the screen?
No.
I'm not sharing that? Uh-oh.
No.
That's another good thing. All right, let's go. Hold on. Let me go back here. Let's try sharing that again. All right. Okay. Can you see it now?
Yes.
Can you
Yeah.
see my mouse now?
Yes.
All right, so let's get rid of all the labels. Oops. There. So here are the concha like we were talking about. Here is the notch they've cut out of that. So normally the inferior nasal concha, as we see, would be a solid structure. But notice that little opening right there, that is the opening where the tears from, again, because if you think of the cyst, this would be the right eye. The right eye would be over here to the side. And those tears drain down that lacrimal duct from a nasolacrimal duct through that opening into the nasal cavity, mixing with the mucous membrane. So that right there, notice they didn't label it on theirs, but that is it right there. And that's what's awesome about this particular half head. This is the half head from one of our torsos. So not only does it have the olfactory nerves drawn in, which is really cool, but it also has the opening for the nasolacrimal duct right there. So that would be it right there. All right. Bring back up my chat window. Where is my chat window? There it is. All right, excellent. Vibrissae, remember, are basically the nose hairs. So, right, next time you're in the bathroom, right, using the rubbery flexible part of your nose, you could elevate your nose, look into your external nares. So when you look inside your external nares, you'll see a bunch of nose hairs. So nose hairs are not what you're going to call them on the test. Instead, you will use the fancy technical term, which are vibrissae. The arytenoid cartilage and corniculate cartilage are what rotate the vocal folds. So if we come back again to the model here, this is that cricoid cartilage that goes all the way around. This is the arytenoid, and these little nubs here are the corniculate. As you can see, there's some cords that are connected to it. They represent the muscles and ligaments. And when they pull, basically they rotate this outward. And as they rotate this outward, that opens up the glottis, the space from the top. So again, this model, we don't have a superior view looking down into it. But right here between them is where the glottis is. And when they're like this, they're closed. But when they rotate out, they swing that open. And as they swing that open, it opens our airway. So basically, the arytenoid and corniculate rotate to open and close the glottis. Keep losing my chat window. Hold on. Come on. There it is. All right. Any more questions? So on the bright side, you guys only have to sit through this twice a week. I have to do this 4 times a week. Exactly. Yeah, I need good luck. Excellent. Auditory station tubes. Those are, we go back to this, although our chart shows it really nicely as well. Let's go with this one. So the same way this is the opening for the tears to come into the nasal cavity. The other space that is open to the nasal cavity, or really to the nasal pharynx, is your middle ear. Your middle ear is an air-filled space where we have those tiny bones, those auditory ossicles that vibrate, and that's how we perceive sound. The way we're able to perceive sound is they vibrate, and for them to vibrate, they have to be in an air-filled space. However, as you have ever known, if you've ever gone up in an airplane or have ever gone, just driven up to Tahoe for the weekend, air pressure changes with elevation. And so your ears pop as a result of that because the pressure inside needs to change as well. And the way we do that is by having a muscular tube that connects from your middle ear to your nasopharynx, which is basically the opening to it right here. So when you yawn or you chew, you move your jaw, that opens that muscular tube and helps to equalize the pressure. So here we see that opening for that structure there. All right. And see that one they have labeled. Eustachian tube is the much more antiquated term. Auditory tube is actually what's called. And again, I'm sure it was Bob Eustachian who first described it. So that's where it got its name. Glad to be of service. Any other questions? The address is on the, if you look in the announcements where I talked about it, you can see it there. But honestly, the other way you can do it is just do a search for Daniel Slutsky in YouTube, and you should be able to get to the channel that way. There was a test one that I did just when I opened up the channel, and then the practice one, which was 3 hours, was on there as well. So either look in the announcements or the email that I sent because the address is there. But like I said, in YouTube, if you just do a search for Daniel Slutsky, I can't imagine there are too many Daniel Slutskys. My picture, I assume, is still my Synapse one. So I think that's what I said is the picture. Excuse me. So you should be able to find it that way. Ah, there you go. Even better. It's always been my hopes and dreams to be YouTube famous. So now it'll happen, hopefully.
I did have a question about the terminal bronchioles.
Yes.
You said that it's a terminal because if it ends there.
It is the conducting zone ends there.
Okay.
So it's the end of the conducting zone. So if I don't think I have they don't have a model that does that. But yes, so it's the terminal because it's the end of the conducting zone. From that point up to that point, everything from the nose to that point, we're just moving and processing the air. However, okay, once it exits the terminal bronchiole into a respiratory bronchiole, then now gas exchange can start to take place.
Okay, and then weren't you saying something about if it has alveoli, then it can do that, but if not, then it doesn't?
Right. So terminal bronchioles do not have alveoli, so that's why they're part of the conducting zone. Respiratory bronchioles do have alveoli, and so that's why they're part of the respiratory zone. That's the only difference between a terminal bronchiole and a respiratory bronchiole. Anatomically, they're the same. They both have simple cuboidal epithelial tissues. They still have a thin, smooth muscle wall, right? They're both about the same size. Literally, the only difference between them is the terminal bronchiole do not have alveoli, So they cannot do gas exchange, so they're just part of the conducting zone. Whereas the respiratory bronchiole will have 1 or 2 alveoli on them, so they can.
Okay. Thank you.
Yep. And to answer the other question, Amy, I'm shockingly new to YouTube from having a YouTube channel. The only reason I would take a video down is if I run out of space. I don't know how much space they allow you to store videos, but as long as they allow, if there's no restriction, then I don't see any reason why I wouldn't leave them up. So yeah, because again, it's a cumulative exam, so we need to eventually know this stuff for the end. So I don't see any reason why we wouldn't have it on there. It might, I guess, eventually it's only half the class, but I guess theoretically down the line it could be useful for people if they miss a day. But again, I don't always say the same things. I'm always constantly tinkering with the lectures, but I guess it's better than nothing. But yeah, definitely for this semester, I will leave them up unless, like I said, unless there's a size or a number restriction, in which case then I will start dropping the other ones. But I will always warn you ahead of time before I do that. Yes. If I become famous, then you can all get a cut of the money. I don't anticipate a huge demand for anatomy and physiology lectures, so I think I'm pretty safe. Of course, I like to see them better. If you want to cut, then you have to ask at least one question during one of the lectures. So that makes it more interactive, forces you guys to talk, and then you guys get to be my supporting cast. Like I said, as you look at this again, if you do look at it again, or as you're looking at your notes, not only if you have any questions, but like I said, this literally is the first time that I've done this. So if there are any suggestions you have for me that can make it run more smoothly, and then obviously the more we do it, hopefully the more comfortable I'll get. Hopefully the volume was good and all those things. I have a special microphone that ironically my daughters use to record their music with. So at least I have a good microphone. Hopefully I have the volume set good on that. So again, please feel free to be critical of this process because like I said, as I've been saying all along, this is new for all of us. I never signed up to be an online instructor. I know none of you signed up to take anatomy and physiology online. So this is new for all of us. So it's going to be a learning process for all of us. So I want to make this the best possible learning experience. So anything you can do to help me be more successful for that is greatly appreciated. Don't forget also, you probably haven't used it or taken advantage of it that much, but remember there's also a discussion board. I honestly haven't looked to see if anybody's on there, but please remember there is a water cooler. There is a discussion board to allow you guys to interact as well. So those are ways that you guys can find ways. Again, now that you have Zoom, you guys can set up your own classrooms and connect to people to you with a free account. And again, please not everybody should have a free account, but with a free account, you can use Zoom to conference with other people. If you have more than 2 people with a free account, you can only do it for 40 minutes. But still, that's a long study session. So again, use that to coordinate and to use your own time so that you guys can meet and do those things that you can do. All right, last call for questions. Any more questions? Comments, concerns, complaints? Yeah, the practice, excellent. Yes, the practice anatomy lab, which is a disc that came in the bundle, but also if you bought one of the more recent bundles, the practice anatomy lab is also accessible through through your modified Master A&P. So when you go into your Master A&P under your study area and lab tools, there is that as well. And so that's really excellent as well. And yeah, if you want to actually send me those slides, I can post them on Canvas as well. So I'm happy to do that as well. If you have some good Google slides that I can, I'll print them as PDFs and post them. But if you want to be able to do that, or like I said, what you can do is also, you guys can set up through Google and use the discussion board to make those arrangements as well. All right, so last call, going once, going twice. Any more questions or comments? All righty, guys, thank you very much. Today's Wednesday, so we've had a grueling one-day work week, but again, I will happily answer questions. I will, I'll be available I haven't figured out how exactly I'm gonna do my office hours yet, but we'll figure something out for that. But definitely by email, I'll be watching my email constantly and can meet with someone through Zoom or anything else if you guys need to do that. Otherwise, I'm going to get this process and get this post. You guys have a great day. Study hard, work hard, and I will see you at lecture on Monday. All right, take care.
Thank you.
All right, bye, guys.
Bye.
Bye.
Bye.